continue with our discussion on the book of Cellular and Molecular Immunology, and today we'll be discussing the chapter number 7, which is Immune Receptors and Signal Transduction. So, in this chapter, on overview, we'll discuss about signal transduction, the modular signaling proteins and adapters, the phases separation of signal protein, the immune receptor families, the general features of antigen receptor signaling, the T receptors complex and T cell signaling. We'll go further and also explain about the B lymphocyte antigen receptor complex, the structure of B cells, receptor for antigen signal initiations, and other things. We'll discuss on immune receptor family. We'll discuss on the T cell receptor complex and T cell signaling with other thing component. Discuss on the B lymphocyte antigen receptor complex. We'll discuss about the attenuation of immune receptor signaling, inhibiting the receptor of natural killer cell, B cell, T cell. We'll discuss on cytokine receptor and signaling, then we'll see the summary. We might end up dividing this pattern or this chapter into two categories, so let us see as we read. So, start with the idea that cells have specific receptor that can be triggered by external ligands came from one of the founders of the modern immunology, the Paul Ehrlich, in his side chain theory published in 1997, conceived of antibodies on the surface of immune cell that recognize antigen and instruct the cell to release more of the same antibody. Cell surface receptor for hormone were discovered many decades later, in the second half of the 20th century, but well before the identification of the antigen receptor on lymphocyte in the early 1980s. Cell surface receptors serve several major functions, including the induction of intracellular signaling, leading to the cell activation and adhesion of one cell to another, or to the extracellular matrix and the internalization of extracellular molecule and cells. Signal transduction broadly refers to the intracellular biochemical pathway that are activated in the cell after the binding of the ligand to specific receptor. Most but not all signaling receptors are located in the plasma membrane. Signaling initiated by these receptors typically involve an initial cytosolic phase when the cytoplasmic portion of the receptor or of the protein that interact with the receptor may be enzymatically modified. This often leads to the activations and or nuclear translocation of transcription factors that are inactive in resting cells. It is followed by nuclear phase when the transcription factor bind to a target DNA and orchestrate the changes in gene expression. Some signal transduction pathways stimulate cell motility or activate granular exocytosis from the cytoplasm without a change in gene expression. Signal transduction can result in a number of different consequences for the cell of the immune system, including commitment to a specific lineage, induction of differentiation, proliferation from the protection from the cell death, initiation of the proliferative response, performance of the effector function, and induction of cell cycle arrest or of the death by apoptosis. Antigen receptors on B and T cell lymphocytes are among the most sophisticated cell signaling machines known, and they are discussed in depth in this chapter. We will first provide a broad overview of signal transduction, followed by discussion of signal mediated by clonal distributed antigen receptor in lymphocyte. When discussing the antigen receptor in T and B cell, we will examine the role of other receptors, including some called co-receptor and other referred to as co-stimulatory receptor, which enhance lymphocyte activation by antigen receptor. We will also discuss the role of inhibitory receptor in T, B, and natural killer cell, NK cell, and consider different categories of cytokine receptor and signal transduction mechanism initiated by these receptors. Finally, to illustrate the step in activation of a prototypic transcription factor, we will examine the pathway that leads to the activation of the nuclear factor kappa B, a transcription factor of relevance to both innate and adaptive immunity. So this is the structure that we are seeing here. So we have the ligands, we have the receptors in the membrane of the cell. Then we see non-receptor tyrosine kinase. We have the inactive downstream enzymes. They can be modified after attachment. You see they are modified. You see there is attachment here, and there is process of phosphorylation happening, modified translocation factor. Then what will happen, you see this is more combined, something is going in so to the nucleus. So there is a phase, a signaling from the cell surface involving cytosolic and nuclear phase, and this was mostly the cytosolic phase, and this one you see it is already in DNA, therefore this is nuclear phase. We'll see more. So reading that the signaling from the surface involves cytosolic and nuclear phase, a generic receptor that activates the non-receptor tyrosine kinase after it binds ligands is shown. In a cytosolic signaling phase, the non-receptor kinase phosphorylates a key tyrosine residue in the cytoplasmic tail of the receptor, as a result of which the phosphotyrosine containing receptor tail is able to recruit a downstream enzyme that is activated once it is recruited. In a cytosolic phase, this activated downstream enzyme post-translation modifies a specific transcription factor that is located in cytoplasm. In this simplified example of cytosolic phase has only a single enzymic event, but many actual signal transduction pathway involve multiple steps. In the nuclear phase, this modified transcription factor enter the nucleus and bind to a specific site in the promoter or in some of the regulator regions of the target genes, and thus facilitate their expressions. So the overview of signal transduction. Receptors that initiate signaling response are generally integral membrane protein present on the plasma membrane, where their extracellular domain recognize soluble secreted ligands or structure that are attached to the plasma membrane of the neighboring cells or to the extracellular matrix. Another category of receptor, nuclear receptor, are intracellular. Both cytosolic and nuclear family member are existing. Transcription factor that are activated by ligand soluble ligand that can cross the plasma membrane. The initiation of signaling from the cell surface receptor may require a ligand-induced clustering of receptor proteins called cross-linking, or may involve a conformational alteration of receptor induced by its association with the ligands. Both mechanisms of signal initiation typically result in the creation of structural change in cytosol proteins protection up or portion of the receptor that promote the recruitment of and or interaction with other signal molecules. A commonly early event in signal transduction is enzymic addition of a phosphate residue on the side chain of amino acid in the cytosolic portion of a receptor or in other proteins that are involved in the signaling. Sometimes a phosphate residue is added to a lipid on the inner leaflet of the plasma membrane. The enzymes that add phosphate group onto amino acid chain are called protein kinase. Many of the initiation events in lymphocyte signaling depend on a protein kinase that phosphorylate the specific tyrosine residue, and these enzymes are therefore called protein tyrosine kinases. Other protein kinases that are involved in a distinct signaling pathway are serine and threonine kinases, which phosphorylate serine or threonine residue. The enzymes activated downstream of signaling receptor that phosphorylate lipid substrate are known as lipid kinase. For the most known phosphorylation event, there are also specific phosphorylate enzymes that can remove phosphate residue and thus modulate signaling. This phosphatase plays an important, usually inhibitory role in the signal transduction. Phosphorylation of protein is not the only post-translation modification that drives a signal transduction. Many of other modifications can facilitate signaling events. A type of modification that we will describe later in this chapter is a covalent addition of ubiquin molecule that either target the protein for degradation or drive signal transduction in many cells, including the lymphocyte. Many important signaling proteins are modified by the addition of lipid that may help localize this protein to a specialized region of the plasma membrane in order for them to efficiently interact to other signaling molecules that are also targeted to this membrane microdomain. Some transcription factors are functionally modified by acetylation, and the N-terminal tail of histones can be acylated and methylated in order to modulate gene expression, DNA replication, and DNA recombination events. So we see these are non-receptor tyrosine kinase based receptor. This is the ligands. Then after that, tyrosine kinase ligands together here receptor. You see the P is involved now, kinase domain. This P is released. After they've binded, then the P is combined, is released. Now, and this is goes to the nuclear hormone. You can see nuclear hormone receptor, the nucleus transcription of nuclear hormones target genes. There's a GPCR ligands can happen again. So when we read this, major category of signaling receptor in immune system depicted here are receptor that uses a non-receptor tyrosine kinase, a receptor tyrosine kinase, a nuclear receptor that bind its ligands and can then influence transcription, a 7 transmembrane G protein coupled receptor. This is GPCR. And notch which are recognized a reagent of distinct cell and is cleaved, yielding intracellular fragment, IC notch, that can enter the nuclear and influence the transcription of specific target gene, ATP, adenosine triphosphate, the cyclic AMP. So cellular receptors are grouped into several categories based on the signaling mechanism they use and the intracellular biochemical pathway they activate. Some receptors use non-receptor tyrosine kinase. The cytoplasmic tail of the ligands binding polypeptide of these receptors have no intrinsic catalytic activity, but a separate intracellular tyrosine kinase known as non-receptor tyrosine kinase participates in receptor activation by phosphorylating specific motif on the receptor or on the protein associated with receptor. Immune receptor constitute a specific family of receptors whose members recognize either antigen or Fc portion of antibodies. All members of this family use a non-receptor tyrosine kinase to initiate a signaling. In addition to the immune receptor family, some cytokine receptor discussed later in this chapter uses non-receptor tyrosine kinases. Integrins, key adhesion receptor in the immune system, may also signal by activating non-receptor tyrosine kinase. So we are having a receptor tyrosine kinase. These are integral membrane protein that activate the intrinsic tyrosine kinase domain or domains located in cytoplasmic tail of a receptor when they are cross-linked to multivariant extracellular ligands. This category of receptor is important in hematopoiesis but does not play a central role in lymphocyte activation. Example of an RTA, this is a receptor tyrosine kinase relevant to blood cell formation, is the c-KIT protein. Other examples of receptor tyrosine kinase include the insulin receptor, the epidermal growth factor receptor, and the platelet-derived growth factor receptor. In vertebrae, a small number of receptors have cytosolic catalytic domain that phosphorylate target proteins or on a serine or threonine residue and not on the tyrosine residue. Two examples will be discussed later in this chapter when we consider a transforming growth factor beta, the transforming growth factor beta signaling. The nuclear receptors are typically located in or migrate into the nuclear where they function as transcription factor. The binding of lipid soluble ligands to its nuclear receptor enable the receptor to stimulate or repress gene transcriptions. Nuclear hormone receptors such as a vitamin D receptor and the glucocorticoid receptor can influence the maturation and activation of immune cells and modulate the cytokines gene expression. We have G protein coupled receptor, a function by activating associated guanosine triphosphate (GTP) binding protein or G proteins. These receptors are polypeptides that transverse the plasma membrane 7 times, because of which they are sometimes called the serpentine receptor or 7 transmembrane receptor, because they are doing this what is called the transverse of the plasma protein 7 times. A conformational change induced by binding of a ligand to this type of receptor permits the activation of an associated heterotrimetic G protein by exchange of bond GTP with GTP. The activated G protein initiates downstream signaling events. Examples of this category of receptors that are relevant to immunity and inflammation include receptor of leukotrienes, prostaglandins, histamine, complement fragments C3A and C5A, bacterial-friendly peptides, sphingosine 1 phosphate, and all chemokines. Different types of G protein linked to distinct GPCR may activate or inhibit different downstream effectors. Two major enzymes that GPCR activate are adenine cyclase, which convert adenosine triphosphate ATP to the effector molecule cyclic adenosine monophosphate, capable of activating numerous cellular response, and phosphatase C, which also trigger multiple signaling events as discussed later. Other classes of receptor have long been known to be important in embryonic development and in certain mature tissue, and their function in immune system have begun to emerge more recently. Receptor protein of Notch family are involved in development in a wide range of species, and the association of specific ligand with the receptor of this family lead to the proteolytic cleavage of receptor and nuclear translocation of cleaved cytoplasmic domain intracellular notch, which function as component of transcription complex. Notch proteins contribute to the cell fate determination during lymphocyte development. I will see it in chapter 8, and may also influence the activation of mature lymphocytes. A group of ligands called WNT protein can influence lymphopoiesis. The names of many proteins involved in signaling are often based on how they were discovered and do not reflect their function, so we will use the general accepted abbreviation and not list of full name. Signaling through a transmembrane receptor of this protein can increase the level of beta-catenin, which can enter the nuclear and activate transcription factors that contribute to the B and T cell development as discussed in chapter 8. Numerous other signaling receptor and pathway first discovered in non-immune cell population are now being studied in context of lymphocyte biology. We will not attempt to comprehensively consider all these pathways in this chapter. So we have the CRP family kinase, SYK family kinase, we have TEC family kinase, we have SH2 domain bind lymphocytes, SH3 domain, and PH domain. So U is a unique domain, T is TEC homolog domain, and K is kinase domain, P is proline rich peptide. Actually, this figure is the modular structure of tyrosine kinase that influence lymphocyte activation. Modules including SH2 domain that bind to the specific phosphotyrosine containing peptides, SH3 domain that recognize proline-rich stretches in polypeptide, PH domain that recognize PIP3 other phosphatidyl nosyl derived lipid, TEC homology domain found in tyrosine kinase of TEC family. Tyrosine kinase family depicted are the SRC family kinase, which include, we have seen there, the CSRC, LIN, FIN, FYN, LCK, the CYK family kinase, which include the CYK, ZAP70, and the TEC family kinases, which include the TEC, BTK, 8K, PH, plectrin homologue, SHSRC homologue. So we'll study more on that. So modular signaling protein and adapters. Signaling molecules are often composed of distinct modules, each with a specific binding or catalytic function. The concept of module signaling molecule has been best illustrated from the study of non-receptor tyrosine kinase. The module structure of several families of tyrosine kinase that are important in the immune system are depicted in figure 7.3. The cellular homologue of the transforming proteins of the rose sarcoma virus called CSRC is the prototype of an immunologically important family of non-receptor tyrosine kinase known as SRC family kinase. The CSRC contains several distinct domains, two of which called SRC homologue 2 and SRC homologue 3 domains mediate binding to other signaling proteins. CSRC also contain catalytic tyrosine kinase domain and N-terminal lipid addition domain that facilitates the covalent addition of mystric acid molecule to the protein. Mystrate help the target SRC family kinase to the plasma membrane. So these are selected adapters that participate in lymphocyte activation. On left, we see the LAT, this one, an integral membrane protein that function as adapter and two cytosolic adapter. The GALT, this one, and also the SLP, this one. So the GAD SLP are shown in a non-covalent T cell. On the right, after T cell activation, what happened? LAT is tyrosine phosphorylated. We want to see the LAT. The LAT, this is what LAT. We see there is a phosphorylation which is happening with these P things. And it is shown to have recruited PLCY or PLC gamma. This one, which facilitates the signaling by calcium, which is simultaneously bind to the membrane phospholipid phosphatidylinositol triphosphate or PIP3, and the GAD adapter, both of which contain SH2 domain, a protein-rich amino acid stretches to SLP76 associated with an SH3 domain of GAD and tyrosine phosphorylation SLP72 recruit valve. Linker for activation of T cell. PH, plectrin homologue. PLC gamma phosphatase SH, SRC homologue. So SH2 domains are composed of about 100 amino acid folded into a particular conformation, and they bind to phosphotyrosine containing peptide in various protein. In antigen receptor signaling, SRC family kinase phosphorylate tyrosine residue present in certain motif in the cytoplasmic tail of protein that are part of receptor complex described later. This phosphotyrosine motif in the antigen receptor complex then serve as binding site for SH2 domain present in tyrosine kinase of SYK family, namely SYK and ZAP70. They recruited SYK or ZAP70 to an antigen receptor by means of a specific SH2 domain. Phosphotyrosine interaction is the key step in antigen-induced lymphocyte activation. SH3 domain are also like about 100 amino acid in length and they help mediate the protein interaction by binding to the proline rich but not phosphorylated stretching certain protein. Another type of modular domain called the plextrin homologue pH domain can recognize specific phospholipid. The pH domain in a number of signaling molecules, including the TEC family, tyrosine kinase, the BTK recognizes phosphatidyl lysosyl triphosphate, PIP3, a lipid moiety on the inner leaflet of the plasma membrane. So adapter proteins function as molecular hubs that physically link the different enzymes and promote the assembly of complex of signaling molecules. These are called adapter protein. Adapters may be integral membrane proteins such as LATs, or they may be cytosolic proteins such as BL and K, SLP76, and GAG. A typical adapter may contain a few specific domains that mediate protein-protein interactions, such as those involved in SH2, SH3 domain. There are many more types of modular domain not mentioned here. Adapter often contain some protein-rich stretches that can bind other proteins that contain SH3 domain. And they are also often containing tyrosine residue that may be phosphorylated by tyrosine kinase and serve as a docking site for other signaling molecule. The amino acid residue that are close to phosphorylated tyrosine moiety determine which specific SH2 domain containing protein may bind to at that site. For example, a tyrosine kinase may phosphorylate a YXXM motif where Y represent tyrosine, M represent methionine, and X refer to any amino acid in an adapter protein, and this will permit binding of an SH2 domain in the lipid kinase phosphatidyllysinol 3 kinase PI3 kinase, but will not recruit other proteins that contain slightly different SH2 domain that are not specific for phosphorylated YXXM motif. A protein-rich stretch in the same adapter protein may bind an SH3 domain in a distinct tyrosine kinase. Thus, tyrosine phosphorylation of adapter can result in tyrosine kinase and PI3 kinase being patched next to each other, resulting in phosphorylation and activation of P13 kinase. Signal transduction can therefore be visualized as a kind of social networking phenomenon. An initial signal, tyrosine phosphorylation, for instance, resulting in protein being brought close to one another and designated hub adapter, resulting in the activation of a specific enzyme that eventually influence the nuclear localization or activity of specific downstream transcription factor or induce other cellular events such as actin polymerization. Phase separation of signaling protein. An underlying principle of signal transduction is the formation of complex of activated signaling molecule reorganized into tiny drops in different phases of matter, much like the formation of droplets of oil in water. The phase-separated protein initiate and amplify and propagate signal efficiently, primarily because they have been brought into the close proximity and at high density. Phase separation is now recognized as a fundamental biological mechanism wherein distinct activated molecule assembles into a different phase from the neighboring constituent of the cell. This process is important in many reactions, including but not limited to signaling and transcription. These include the assembly of adapter after T cell receptor signaling, the activations of cytosolic receptor for nucleic acid, and formation of inflammasomes. The immune receptor family. Immune receptor are a unique family of receptor complex typically made up of integral membrane protein of immunoglobulin superfamily that are involved in ligand recognition associated with other transmembrane signaling protein that have a unique tyrosine containing motif in their cytoplasmic tail. Whereas the signaling component are generally distinct from the protein involved in the ligand recognition, in a few members of the family, the receptor consists of a single chain in which the extracellular domain is involved in ligand recognition and a cytoplasmic tail containing tyrosine residue that contribute to the signaling. The signaling protein of the immune receptor family are often positioned close to a non-receptor tyrosine kinase of the SRC family, which possess N-terminal lipid anchor that tether them to the inner leaflet of the plasma membrane. The cytoplasmic tyrosine containing motif on the signaling proteins of the immune receptor family are generally one of the three different types, one being an activated motif and the other inhibitory and other third can either activate or inhibit depending on the cell types and the particular immune receptor. Immunoreceptor tyrosine-based activation motif items are found in receptor and associated protein involved in cell activation and have the sequence of YXXL, 1, X bracket 6 to 8, YXXL stroke I, where Y represent a tyrosine residue, L represent leucine, I represent isoleucine, and X refer to any amino acid. Both tyrosine residue in item, the immunoreceptor tyrosine-based activation motif, can be phosphorylated by SRC family kinase when immune receptor are activated by the ligands. Tyrosine phosphorylated items recruit either SYK or ZAP70, which contain tandem SH2 domain that each bind to one of the two phosphorylated YXXL-1 I motif of the item. Binding of SYK or ZAP kinase to the phosphorylated item causes a conformational change that activates the kinase, leading to additional signaling events that drive the immune cell activation. Some immune receptor inhibit cellular response, and signaling chain in these receptor may contain a slightly different tyrosine containing motif that is called an immune receptor tyrosine-based inhibitory motif item, not item, which has a consistent sequence of V stroke L stroke I X Y X X L, where V refer to valen. Phosphorylated items recruit tyrosine phosphatase or inositol lipid phosphatase enzyme that remove phosphate residue from phosphotyrosine moieties or from the certain lipid phosphate and thus counteract the item-based immune receptor activation. Certain receptors contain a cytosolic motif called immunoreceptor tyrosine-based switch motif. ITSM that has a consensual sequence of a TXY, XXV, stroke I. This motif can sometimes function in an inhibitory function and recruit the SH2 domain containing tyrosine phosphatase. As in the case of ITSM, in the cytosolic tails of programmed cell death, PD1, it is called a switch motif because in some receptors of the SLAM family of the instance, this motif can orchestrate a switch from the binding of tyrosine phosphatase SHP2 to binding a tyrosine kinase such as FYN, depending on the absence of or presence of respectively of adapter called SAP, SLAM associated protein. Thus, the ITSM can mediate a change from an inhibitory to an activating function. Members of immune receptor family include antigen receptor on the B cell and T cell, Fc receptor on the B cell, myeloid cell and mast cell, and activating an inhibitor receptor on natural killer cell, T cell, and B cell. The recognition protein of the several activating receptor in the immune system do not have a signal motif in their cytoplasmic tail, but signal by forming a complex with item containing protein that contribute to the signal transduction from receptor complex. These signaling protein include Zeta Chain and CD3 proteins of the T cell receptor complex, IgA alpha and Ig beta, Ig alpha and Ig beta protein associated with the antigen receptor of B cell and component of several Fc receptor of the NKG2D activating receptor on natural killer cell. Many inhibitory receptor, including CD22 on B cell, Fc gamma RIIB on B cell and other cell, and several inhibitory natural killer cell receptor containing items in their cytoplasmic domain. The T cell inhibitor receptor PD1 containing items motif as well as items in its tail. So we see this. Selected members of immune receptor family. 5 selected members of immune receptor family are depicted. Typical immune receptor that activate this immune cell have separate polypeptide chain. For recognition and associated polypeptide chain that contain cytolytic items. Examples shown here include B cell receptor and T cell receptor. And the high affinity receptor for IgE, the Fc epsilon RI inhibitor receptor in the immune system typically have items on the cytosolic portion of the same chain that uses its extracellular domain for ligand recognition. Fc gamma RIIB is an inhibitor receptor found on B cell and myeloid cell. PD1, an inhibitor receptor found on T cell, also has immunoreceptor tyrosine based switch of motif, ITSS, in its cytosolic tail. Ig, immunoglobulin, items, immunoreceptor, tyrosine based activation motif, items, the other things. So these are what we see in here. We have the Ig beta alpha, this is B cell receptor. We have CD3 alpha epsilon, this is T cell receptor, CD3 epsilon delta, these are beta. These are the inhibitor receptor. This is for T cell, this is for B cell, and this is for B cell, and it also works for myeloid cells. Yes. General features of antigen receptor signaling. Signaling downstream of T and B cell antigen receptor is characterized by a similar sequence of events consisting of the following. A receptor ligation typically involves the clustering of receptor by multivariant ligands and result in activation of an associated SRC family kinase. In some cases, SRC kinase is brought into proximity with the receptor and associated proteins. Receptor ligations may also induce the unfolding of cytoplasmic tail of the polypeptide chain that is part of the receptor. This conformational change may allow previously hidden tyrosine residue of the cytoplasmic item to become available for phosphorylation by SRC family kinase. The activated SCRC family kinase phosphorylate available tyrosine in the items of signaling protein that are part of receptor complex. 2 phosphorylated tyrosine in a single items are recognized by SYK or ZAP70 kinase, which contain a tandem SH2 domain that each bind to the item phosphotyrosines. Recruitment of SYK or ZAP70 to phosphorylated items result in a and the activation of kinase and subsequent tyrosine phosphorylation of multiple adapter protein and enzymes that activate distinct signaling pathway downstream of the immune receptor. This sequence of events is described in more detail in the context of T-cell receptor and B-cell receptor signaling later in the chapter. Alteration in the strength of T-cell receptor and B-cell receptor signaling influence the response of lymphocytes during their development and activation. In other words, the presence of different number of activated signaling molecules induced by antigen-ligated receptor is interpreted differently by lymphocyte. For example, during lymphocyte development, weak antigen receptor signaling is required for survival of clone expressing the functional receptor positive selection. A strong signaling is required to induce apoptosis of clone with self-reactive antigen receptor negative selection. In mature lymphocytes, strong signaling usually results in a clonal expansion and differentiation of naive lymphocyte and the acquisition of the host defense function by effector lymphocyte. Antigen receptor signaling is modulated by mechanisms that are unique for this class of receptor. Several such mechanisms have been described. The progressive item use. One of the way in which a quantity of signal output from antigen receptor might be altered is phosphorylation of different number of items tyrosine after receptor engagement. The T-cell receptor complex has 6 signaling channels and 10 items, and increasing number of items may be phosphorylated with a strong or propagate prolonged binding of antigen to T-cell receptor. The number of items phosphorylated may therefore provide a cytosolic interpretation of the strength of antigen binding to the T-cell receptor, and this can influence the nature of cellular response. The B-cell receptor has only 2 items, but because this number increases when multiple B-cell receptors are cross-linked by multivariant antigen, the degree of cross-linking by antigen may determine the number of items that might be used and thus generate different response to antigen of different affinity and valence. Increase the cellular activation by co-receptor. A co-receptor is a transmembrane signaling protein on a lymphocyte that can facilitate antigen receptor activation by simultaneously binding to the same antigen complex that is recognized by antigen receptor. The co-receptor brings with its signaling enzymes linked to its cytoplasmic tail and can thereby facilitate item phosphorylation and activation of antigen receptor when antigen draw into the vicinity of antigen receptor. Co-receptor on T cells are the CD4 and CD8 protein that demarcate two functionally distinct subsets. Complementary receptor type 2 complementary receptor 2 comes to result 21 is the receptor on the B cells. Modulation of signal by inhibiting receptor. A key inhibitor receptor in T cell include CTLA4 and PD1, and the important inhibitor receptor in B cell are CD22 and Fc gamma RIIB. Among others, the role of these inhibitors are discussed later in this chapter. In addition, antigen receptor signal may, in some circumstances, cooperate with signal from protein called costimulatory receptor that add yet another level of control to process of lymphocyte activation. This process is best documented by T-cell activation. Costimulatory receptor provides so-called a second signal for lymphocyte. Antigen recognition provides the first signal and ensure the immune response is optimally triggered by infectious pathogen and substance that mimic microbes, which are the agent that induces or activate the costimulator. Unlike co-receptor, co-stimulatory receptor do not bind to the antigen that are recognized by antigen receptor, but rather to entirely distinct ligand on antigen presenting cell that are induced by pathogen and other potentially dangerous insult. Signal output downstream of costimulatory receptor cooperate with signal derived from antigen receptor antigen receptor to full activate lymphocyte. The prototypical costimulatory receptor is CD28 on T cell, which is activated when bound to costimulatory molecule B71 CD80 and B2 CD86 expressed on antigen presenting cell. This discussion of general principle of signaling in the immune system provide the basis for considering in more detail specific pathway activated in T and B cell by receptor force antigen entry stimulators. The T cell receptor complex and T cell signaling. The T cell receptor was discovered in the early 1980s at around the same time that the structure of the major histocompatibility complex molecule with bound peptide, the signal for T cell, was being defined. This was a year after B cell antigen receptor and Ig genes were characterized. The method used to search for protein of the T cell receptor and the gene encoding them relied on the assumption that they would be similar to Ig protein and genes. We now know that all T cell receptors are similar to antibodies, but there are also important difference between these two types of antigen. Let us see the table 7.1. Properties of lymphocyte antigen receptor. So we have the T cell receptor and the immunoglobulin G. A component in T cell receptor you have alpha and beta chain, most common. In immunoglobulin you have heavy and light chain. When we're speaking of alpha and beta chain, most commonly form of T, this is for T cell receptor. Number of Ig domain for T cell receptor, 1V domain and 1C domain in each chain. For immunoglobulin, the heavy chain, 1V domain, 3 or 4C domain. Then in light chain, 1V domain and 1C domain. Number of CDRs involved in antigen bindings, here is 6, 3 in each chain. But here in immunoglobulin there are 6, 3 in each chain. Associated signal molecules, CD3 and beta, and here for immunoglobulin is Ig alpha and Ig beta. Affinity for antigen is for T cell is 10 to 10 power negative 7 to 10 power negative 5 to 10 power negative 7. While for immunoglobulin is 10 power negative 7 to 10 power negative 11. Sorry. Changes after cellular activation, production of secreted form. There's no secreted form for T cell, but there are secreted form in immunoglobulin. Isotope switching, no isotope switching in T cell, but there is isotope switching in B cell. Somatic mutation, there is no somatic mutation in T cell, but there is somatic mutation in B or immunoglobulin G. The T cell receptor for antigen. The antigen receptor for MHC restricted CD4 plus helper T cell and CD8 cytotoxic T lymphocyte is a heterodimer consisting of two transmembrane polypeptide chain designated T cell receptor alpha and beta covalently linked to each other by disulfide bridge between extracellular cysteine residue. T cell expressing this form of T cell receptor are called alpha beta T cell. A less common type of T cell is composed of T cell gamma and delta, and the cell on which it is expressed are called the gamma delta T cell. Each T cell alpha and beta chain consists of one Ig, like any terminal variable V chain, V domain, one Ig like constant C domain, a hydrophobic transmembrane region, and short cytoplasmic region. Thus, the extracellular portion of a T cell receptor alpha beta heterodimer is structurally similar to antigen binding fragment. of an Ig molecule, which is made up of the V and C region of the light chain and V region and first C region of heavy chain. So this is the structure of the T cell receptor, the schematic diagram of alpha beta T cell. Left, you see this one, and show the domain of a typical T cell receptor specific for peptide major histocompatibility complex. The antigen binding portion for T cell receptor is formed by V beta and V alpha domain. The ribbon diagram, right, show the structure of the extracellular portion of T cell receptor revealed by X-ray crystallography. The hypervariable region loops, so you see these ones, they are seen, and this is the transmembrane region with the T cell receptor. This is a disulfide bond, Ig domain, carbohydrate, and other things. So form of peptide binding site at the Ig immunoglobulin, this they just defined it. Okay. So this structure here is explained down here. This is the binding of T cell receptor to peptide, major histocompatibility complex. The V domain of T cell receptor are shown interacting with human class 1 MHC molecules, HLA A2 representing a viral peptide in yellow, this one, and the figure represents a frontal view of X-ray crystal structure of the molecular MHC peptide T cell receptor complex with beta 2M, beta 2 microglobulin, HLA, human leukocyte antigen. As you see, this is just a peptide, and this is a T cell receptor, and T cell receptor, here we see there is alpha and beta. This is MHC. The MHC is presenting this thing here to the T cell receptor for other function to happen. So the V region of the T cell receptor and alpha and beta chain contain short stages of amino acid where the variability between different T cell receptor is concentrated, and this form of hypervariable or complementary determining regions, 3 complementary determining region in the chain and 3 similar regions in the beta chains together form the part of T cell receptor that specifically recognize peptide MHC complex. Each T cell receptor chains, like Ig, heavy and light chain, is encoded by multiple gene segments that are joined together during the maturation of T lymphocytes. The C region of both alpha and beta chains continue into a short hinge region which contain a cysteine residue that contribute to the disulfide bond linking to 2 chain. Each hinge is followed by hydrophobic transmembrane portion and carboxyl terminal cytoplasmic tail that are 5 to 12 amino acid long. Like membrane IgM on B cell discussed later, the cytoplasmic region are too short to transduce signal and other protein with item motif that are physically associated with the T cell receptor serves as signal transducing function of this antigen receptor complex. The CD3 and Zeta proteins are non-covalent associated with T cell receptor alpha and beta heterodimer to form a T cell receptor complex, and then the T cell receptor recognized antigen. These associated protein transduce the signal that lead to the T cell activation. The component of a T cell receptor complex are illustrated in figure 7-8. The CD3 protein and the Zeta chains are identical in all T cells regardless of specificity, which is consistent with their role in signaling and not in antigen recognition. The CD3 and the Zeta protein also are required for surface expression of complete receptor complex on T cell. CD3 gamma and delta and epsilon protein are homologous to one another. This N-terminal extracellular region of alpha, delta, and epsilon chain of CD3 each contain a single Ig-like domain, and therefore these 3 proteins are members of Ig superfamily. the transmembrane segment of all three CD chains, and the Zeta chain contain a negatively charged aspartic acid residue that bind to positively charged lysine and adenine residue in the transmembrane domain of T cell-like receptor alpha and beta chains, thus keeping the T cell receptor CD3 and zeta associated as complex. Each T cell receptor complex contain one T cell receptor alpha beta heterodimer associated with the CD3 gamma epsilon heterodimer, one CD3 delta epsilon heterodimer, and one disulfide linked zeta zeta homodimer. To repeat, the transmembrane segment of all three CD3 chains and the Zeta chain contain a negative charged aspartic acid residue that bind to positively charged lysine and adenine residue in transmembrane domain of T cell receptor alpha and beta chain, thus keeping the T cell receptor CD3 and zeta associated as complex. Gamma, the negative and positive. Each T cell receptor complex contains one T cell receptor alpha beta heterodimer associated with one CD3 alpha epsilon heterodimer, one CD3 delta epsilon heterodimer, and one disulfide linked zeta homodimer. The cytoplasmic domain of CD3 alpha, delta, and epsilon protein range from 44 to 81 amino acid residue in length, and each of these domains contain one item. The Zeta chain has short extracellular region of 9 amino acid, a transmembrane region containing a negatively charged aspartate acid residue similar to the CD3 chains, and long cytoplasmic region 113 amino acid that contain 3 items. The Zeta chain is normally expressed as the homodimer, and it is also associated with signal receptor on lymphocyte other than T cell, such as Fc gamma receptor, Fc gamma RIII of the natural killer cell. Ligation of a T cell receptor by MHC peptide ligand results in the castling of the co-receptor with the antigen receptor and phosphorylation of item tyrosine residue in CD3 and zeta protein. In addition, recognition of peptide MHC complex by T cell receptor may induce conformational change in T cell receptor, making the item associated with the linked CD3 or zeta chain available for tyrosine phosphorylation of co-receptor associated SR family kinase. In addition to toll-like receptor complex, T cell expresses several other proteins that recognize ligand on antigen presenting cell and play important role in T cell response. Role of the CD4 and CD8 co-receptor in T cell activation. CD4 and CD8 are T cell co-receptors that bind non-polymorphic region of MHC molecule and facilitate signaling by T cell receptor complex during T cell activation. Mature alpha beta T cell expresses either CD4 or CD8, but not both. CD8 and CD4 interact with class 1 and class 2 molecule respectively and are responsible for class 1 and class 2 MHC restriction of these classes of T cell. See in chapter 6, we discussed already. So we have extracellular space, we have the CD3, these are just markers which are available in a T cell. We have T cell receptor alpha and beta chain. Then we have epsilon, that is 2, we always say they are heterodimer. So as we had above that we read, we have alpha gamma, we have epsilon gamma, we have epsilon delta, but we have also homodimer zeta. The common T cell receptor complex, the T cell receptor complex of major histocompatibility complex restricted T cells consisting of alpha beta T cell non-covalent linked to the CD3 and alpha proteins, zeta proteins. The association of these proteins with one another is mediated by charged residue in their transmembrane region. Remember the negative and positive. So, CD4 and CD8 are transmembrane glycoprotein members of Ig superfamily. CD4 is expressed as monomer on the surface of peripheral T cell and thymocyte, and it is also present at a lower level on mononuclear phagocytes and some dendritic cell. CD4 has 4 extracellular Ig-like domain, hydrophobic transmembrane region, and a highly basic cytoplasmic tail 38 amino acid long. 2 N-terminal Ig-like domain of the CD4 protein bind to the non-polymorphic alpha 2 beta 2 domain on the class 2 MHC molecules. The human immunodeficiency virus, HIV, uses the CD4 as the receptor to gain entry to the T lymphocyte and the immune cell that express and other immune cells that express the molecule. We see in chapter 21. The most CD8 molecule exists as disulfide linked heterodimer composed of the 2 related chain called the CD8 alpha and CD8 beta. Both of the alpha and beta chains have a single extracellular Ig domain, a hydrophobic transmembrane region, and highly basic cytoplasmic tail that is about 25 amino acid long. Activated T cell as well as alpha delta T cell can express CD8 alpha alpha homodimers. The Ig domain of CD8 bind mainly to the non-polymorphic alpha 3 domain of the class 1 MHC molecules and also interact with the portion of alpha 2 domain and with beta 2 microglobulin. The SRC family kinase LCK is non-covalent bound to the cytoplasmic tail of co-receptor CD4 and CD8. The ability of extracellular domain of this co-receptor to bind to MHC molecule on the antigen-presenting cell draw these proteins adjacent to T cell receptor that contact the same MHC molecule that is displayed as peptide on antigen-presenting cell. As a result, on the cytoplasmic phase of plasma membrane, the LCK is brought in close proximity to items in CD3 and Zeta protein. The LCK then phosphorate the tyrosine residue in these items, thus facilitate the subsequent recruitment and activation of ZAP70 tyrosine kinase. Note that LCK is already attached to both CD4 and CD8 co-receptor and is active even before the antigen exposure. The other protein in T cell receptor complex are CD3 and Zeta chains, items that first need to be phosphorylated before they can recruit a kinase. Thus, the co-receptor provides the earliest enzymic activity for initiating signal after T cell recognition of the peptide MHC complex. So these are some of the members, and the ligand receptor pair involved in T cell activation. A, the major surface molecule CD4 cell T involved in the response of this cell, the receptor, and the molecule of antigen presenting cell ligands recognized by receptor are shown. CD8 T cell use most of the same molecule except that T cell receptor recognize peptide class 1 MHC more complex, and the co-receptor is CD8, which recognize class 1 MHC. Immunoreceptor tyrosine based activation motif item are the region for signaling protein that are phosphorylated on the tyrosine residue and become docking site for the other signaling molecule. CD3 is composed of 3 polypeptide chain named alpha, delta, and epsilon arranged in 2 pair, alpha, gamma epsilon and delta epsilon, as shown in figure here, in figure 7.8. Some inhibitory receptors such as PD1 contain cytoplasmic immunoreceptor tyrosine based inhibitory motif item and switch motif immunoreceptor tyrosine based switch motif. Important molecules of T cell that participate in activating inhibitory receptor to antigen but are not receptor for antigen are summarized as these as antigen presenting cell, and here we are. So receptor of CD4 helper T cell, we saw this is CD4, signal transduction, antigen recognition. This is T cell receptor. We have signal transduction. These are the one conducting signal transduction. The CD3 epson. We have inhibition for CD4 is CTLA, for PD1. Adhesion is LF1. Then items ligands of class 2 MHC expressing antigen, they have peptide. This can be class 2. And other ligands are B71, which is 80, B71, B72, which is 80, stroke CD80, CD86, PDL1, this is the receptor for PDL, and this adhesion molecule is the receptor here is ICAM. So B, this is T cell molecule function, ligands. So like we have CD3, the function is signal transduction to T cell complex, and ligands don't have ligands. We have the zeta, it works for signal transduction, but there's no ligands. We have CD4, this works for signal transduction. It is ligand is a class 2 MHC and expressed on antigen presenting cell. We have this ligand is expressed in antigen presenting cell, the ligand which is MHC class 2. CD8, it works for signal transduction. It is on T cell, remember, and ligand is class 1 MHC, and all nucleated cells have them. And CD28, this signal transduction, it is in T cell. It works for signal transduction. Its ligand is B71, B72, which is equivalent to CD8, CD86. These are found in antigen presenting cell. We have CTLA4. These are Wakers inhibitor, and they bind to the same receptor as CD28. They are present also in antigen presenting cell. We have PD1. These are inhibition. These ones are found in T cell, and in response to that we have the PD ligand 1, PD ligand 2. These are also present in the antigen cell, antigen presenting cell. We have LF1. This is for adhesion, and this one ICAM is found in antigen presenting cell. So in summary, most of the ligands are present in antigen presenting cell, and except for the one which is present in nucleated cell, except the red blood cell, I think here. So this is very nice summary to explain what is happening. So this is again a schematic view of structure of CD4 and CD8 co-receptor. And cellular protein is the integral membrane of monomer consisting of 4 extracellular immunoglobulin, Ig domain, a transmembrane domain, and cytoplasmic tail. The CD8 protein is either a disulfide-linked alpha-beta integral membrane heterodimer or disulfide-linked alpha-alpha homodimer, not shown. CD4 T cell associated with the invariant portion of the major histocompatibility complex, class 2, heterodimer on the antigen presenting cell that is interacting with T cell receptor on the surface of T cell. Note that the cytoplasmic portion of both CD4 and ASOS can associate with LCK. Activation of tyrosine kinase and a lipid kinase during T cell activation. Phosphorylation of protein and lipid play a central role in transduction of signal from the T cell receptor complex and co-receptor. Within seconds of T cell receptor ligation, LCK phosphorylate the items of CD3 and Zeta chains. The tyrosine phosphorylated item in the Zeta chains are docking site for SYK family tyrosine kinase called ZAP70, Zeta associated protein of 7th decade. ZAP70 containing 2SH2, domain that can bind to item phosphotyrosine. As discussed earlier, each item has two tyrosine residue, and both of these must be phosphorylated to provide a docking site for one ZAP molecule. The bound ZAP70 become a substrate for adjacent LCK after T-cell receptor recognition of antigen. LCK phosphorylated specific tyrosine residue of ZAP70. As a result, ZAP70 acquire its own tyrosine kinase activity and is then able to phosphorylate a number of other cytoplasmic signaling molecules. A critical threshold of ZAP70 activity may be needed before downstream signaling event will proceed, and this threshold is achieved by recruitment of multiple ZAPs P70 molecule to the phosphorylated items on Zeta chains and C3 tail. Another signaling pathway in T cell involves the activation of PI3 kinase. This enzyme is recruited to adapt a protein associated with T cell receptor complex and phosphorylate phosphatidyl nosyl biphosphate located in the inner leaflet of the plasma membrane to generate PIP3. Certain signaling protein in the cytosol have specialized pH domain that have an affinity for PIP3, and as a result, pH domain containing protein can bind to the inside of the cell membrane only when PIP3 is generated. For example of pH domain containing protein include TEC family tyrosine kinase such as ITK in T cell and BTK in B cell, as well as phospholipase C gamma 1, PLCY1, a key enzyme in the calcium signaling pathway in T cell. Another important PIP3 dependent kinase is PDK1, a serine threonine kinase that is required for phosphorylation and activation of an important downstream kinase called AKT. Activated AKT phosphorylate crucial target and contribute to both increased protein synthesis and metabolism, leading to cell growth, enlargement, and survival. AKT activate another serine threonine kinase called mechanistic target of rapamycin. It is controller of protein synthesis and cell growth. AKT also promotes cell survival in a number of ways, including inactivation of pro-apoptotic protein and increased production and activation of anti-apoptotic protein of BCL2 family. Recruitment and modification of adapter proteins. Activated ZAP70 phosphorylate several adapter proteins, making them capable of binding to signaling molecule. A key early event in T cell activation is the ZAP70 mediated tyrosine phosphorylation of adapter proteins such as SLP70 and LAT. Phosphorylated LAT directly bind to PLC1 discussed later and coordinate the recruitment of several other adapter protein including SLP76, GAD, and GRB2 to the cluster of T cell receptor and T cell receptor associated proteins, sometimes referred to as T cell receptor signal losome. Thus, LAT serves as to bring downstream component of T cell receptor signal pathway close to their upstream activators. Because the function of many of these adapters depend on their tyrosine phosphorylation by active ZAP70, only antigen recognition, the physiologic stimulus for ZAP70 activation, trigger the signal transduction pathway that lead to the functional T cell response. So we have the diagram here. This is T cell receptor complex. Core receptor are clustered within the membrane, lipid wrapped by antigen recognition. We have the CD4, CD8 T cell, then it is CD3, the membrane. This is the antigen presenting cell. So the antigen presenting cell is bringing in the peptide. So this is T cell. The T cell is having CD3 it having CD4 or it can be CD8. Then there's a phosphorylation taking place here. This is making this bind. Then ZAP bind to phosphotyrosine and the phosphorylate adapter. As you see here, what is happening? ZAP is binding, and the latter again. Assemble of adapter proteins. There is assembling of adapter protein. All this that you see. Activation of calcitonin F2 pathway. Activation of RAS pathway. Early tyrosine phosphorylation event in T cell activation. On antigen recognition, there is clustering of T cell receptor complex within co-receptor CD4. In this case, CD4 associated LAC become active, and phosphorylate tyrosine in the item of CD3 and alpha chain. ZAP7 bind to the phosphotyrosine of the chain, and it is itself phosphorylated and activated. The illustration shows one ZAP70 molecule binding to two phosphotyrosine of one item in the chain, but it's likely that initiation of T cell response require the assembly of multiple ZAP70 molecules on items of the two chains as well as CD3 chains. Active ZAP then phosphorylates tyrosine on various adapter molecules such as ALAT. The adapter become docking site for cellular enzymes such as PLC gamma 1 and GDP, the exchange factor that activate RAS and other small G proteins upstream of MAPK. These enzymes activate various cellular response, GDP, guanosine diphosphate, GDP, guanosine triphosphates. So we see this role of PK3 in T cell response. PK3 generated by PI3K activate the PDK1, which phosphorylates they activate the AKT kinase, which in turn phosphorylate downstreams. So please, I would encourage you to have your time to read this through this pathway yourself to understand on how these things works. Formation of the immune synapse. When T cell receptor complex recognize MHC molecule, MHC associated peptide on the antigen presenting cell, several T cell surface protein and intracellular signal molecule are rapidly mobilized to site of T cell antigen presenting cell contact. This region of physical contact between T cell and the antigen presenting forms a bull-eye-like structure that is called immune synapse, a supramolecule activation cluster. The T cell molecule that move to the center of synapse include T cell receptor complex. This is T cell receptor CD3 and Zeta Chain, CD4 or CD8 co-receptor, receptor for co-stimulators such as CD28, enzymes such as PKC Zeta 0, and adapter protein that associate with the cytoplasmic tail for the transmembrane receptor. At this region of the synapse called the CSMAC for central SMAC, the distance between plasma membrane and T cell and antigen presenting is about 15 nanometer. Integrin remain at the periphery of the synapse where they function to stabilize the binding of the T cell to antigen presenting cell, forming the peripheral portion of the SMAC called the PSMAC, the peripheral SMAC. This outer part of the synapse, the two members are about 40 nanometer apart. Many signaling molecules found in synapse are initially localized to region of the plasma membrane that have a lipid content different from that of the rest of the cell membrane and are called rift-raft glycoprotein, glycolipid-enriched microdomains. T-cell receptor and costimulatory receptor signaling is initiated in these rafts, and signaling initiates cytoskeletal rearrangements that allow raft to coalesce and form the immune synapse. Immune synapse serves a number of functions during and after T-cell activation. The synapse form a stable contact between an antigen-specific T-cell and antigen-presenting cell, displaying that antigen and become the site for assembly for signaling machinery for T-cell, including the T-cell receptor complex, co-receptor, costimulatory receptor, and adapter. Although some T-cell receptor signal transduction is initiated before the formation of synapse and is in fact required for synapse formation, the immune synapse itself provides a unique interface for T-cell receptors triggering. T-cell activation need to overcome the problem of generally low affinity T-cell receptor for peptide MHC ligands and the presence of few MHC molecules displaying any one peptide on the antigen presenting cell. The synapse represents a site at which repeated engagement of T-cell receptor can be sustained by this small number of peptide MHC complex on the antigen presenting cell, thus facilitating a prolonged and effective T-cell signaling. The synapse ensures the specific delivery of secretory granules, content, and cytokines from a T cell to antigen presenting cell or to a target that are in the contact with T cell. Vector delivery of secretory granule containing porphyrins and granzymes from the cytotoxic T lymphocyte to target cell occur at the synapse. Similarly, CD40 ligands CD40 interaction are facilitated by accumulation of these molecules on T cell and antigen presenting cell. Interaction interface of the immune synapse. Some cytokines are also secreted in a direct manner into the synapse cleft from where they are preferentially delivered to the cells that is displaying antigen to the T lymphocyte. This ensures that cytokine act on cell-bearing foreign, e.g. microbial antigen, which are the cells that need to be activated or killed to eliminate these antigen. The synapse, especially the cCAM region, also may be important site for turnover of signal molecule, primarily by ubiquination and delivered to the late endosomal and lysosomes. This degradation of signaling protein contributes to the termination of T cell activation and is discussed later. So, map kinase signaling pathway in T lymphocyte. So before we read this, let us see this structure. What does it mean? So here they are speaking of the immune synapse. A is an immune synapse. So this is an immune synapse, A. And actually, immune synapse, this figure shows the view of immunological synapse in a T cell antigen presenting conjugate shown as an Nomex image in parallel C, telling a protein that encoded with a cytoplasmic tail was revealed. I wish to say for B. B, a schematic view of synapse showing taline and LFA1 in the PSMAC green and PKC zeta and a T cell receptor CMAC. So when you see this one, you see this is the synapse that they were narrating. This is where they meet the antigen presenting cell with the MHC class 2, for example, for CD2, for CD4, and this is the T cell. So it has the PKCO There is a receptor here, the toll T cell receptor. There is an antigen here or protein. These all they bind together to make sure that this process is working. So let us continue with MAP kinase signaling pathway in T cell lymphocyte. Small guanine nucleotide binding protein, G protein, activated by antigen recognition stimulate at least 3 different mitogen activated protein kinase, which in turn activate distinct transcription factor. So G protein are involved in diverse activation responses in different cell types. Two major members of this family activated downstream of T cell receptor are RAS and RAC. Each activates a different transcription factor, and together they mediate many cellular response of T cell. You will have to read this one, the structure. The RAS pathway is triggered in T cell after T cell ligations, leading to the activation of extracellular receptor activation kinase, extracellular receptor activated kinase, ERK, a prominent member of MAP kinase family, and eventually to activation of downstream transcription factor. RAS is loosely attached to the plasma membrane through covalent bond lipid. It is in active form. The guanine nucleotide binding site of RAS is occupied by guanosine diphosphate. Upon activation of the cell, the bound GDP is replaced by guanosine triphosphate, and RAS undergo conformational change that can then recruit or activate various cellular enzymes, the most important of which is RAF. The activation of RAS by GDP or GTP exchange is seen in response to the engagement of many types of receptor in many cell population, including the T cell receptor complex in T cell. Non-mutated RAS protein are active GTPs that convert the GTP bond to RAS into GDP, thus returning the RAS into normal inactive state. Mutated RAS protein that are constitutively active, they constantly assume the GTP bond conformation, often because of loss of GTPase activity, contribute to the neoplasmic transformation of many cell types. The mechanism of RAS activation in T cell involve adapter protein LAT and the GRB2. When LAT is phosphorylated by ZAP70 at the site of T cell receptor clustering, it serves as docking site for SH2 domain of GRB2. Once attached to LAT, GRB2 recruit the RAS GTP exchange factor called source to the plasma membrane. Source catalyze the GTP for GTP exchange on RAS. This generates the GTP bond form of RAS written as RAS GTP which then trigger the cascade of the kinase activation. The RAS GTP directly activates a kinase called RAF, the first kinase in this cascade. The RAF then phosphorylates and activates the dual-specific kinase called MEK1, which in turn phosphorylates the third kinase in the cascade called ERK on the closely spaced threonine and tyrosine residue. ERK is the MAP kinase, and MEK1 is called MAP kinase that activate a MAP kinase. The activated ERK translocate to the nucleus and phosphorylate a protein called ELK, and phosphorylated ELK stimulates the transcription of FOS, a component of activation protein 1, AP1 transcription factor. In parallel with the activation of RAS through recruitment of GRB2 and source, the adapter phosphorylated by TCR associated kinase also recruit and activate the GDP exchange protein called VAV that act on RAS, another small G protein. The RAC GTP that is generated initiate a parallel MAP kinase cascade resulting in the activation of distinct MAP kinase, C-Jun, N-terminal kinase, J and K. J and K is sometimes called stress activating protein, SAP kinase, because in many cells it is activated by various noxious stimulus. Activated GK then phosphorylated Jun, second component of UP transcription factor, a third member of the MAP kinase family in addition to ERK and J and K in P38, and it is activated by RAS-GTP and in turn activate various transcription factor. RAS-GTP also induces cytoskeletal reorganization and may play a role in clustering of T-cell receptor complex, co-receptor, and other signaling molecule into the synapse. The activities of ERK and J and K are eventually shut off by action of dual-specific protein, tyrosine, threonine, and phosphatase. These phosphatases are induced or activated by ERK and J and K themselves, providing a negative feedback mechanism to terminate T-cell activation. Calcium and protein kinase C mediated signal pathway in T lymphocyte. T-cell receptor signaling lead to the activation of gamma-1 isoform of enzyme phospholipase C, PLC gamma-1, and provide a PLC gamma-1 mediated hydrolysis of membrane lipid activate additional signaling event that induce specific transcription factor in T-cell. Soon after T-cell triggering, the LAT and SLP76 adapter are phosphorylated on the tyrosine residue by ZAP70 and form a complex on the inside of the plasma membrane. At the same time, PI3 kinase generates PIP3 in the inner leaflet of the plasma membrane and recruit a TEC family kinase ITK and also PLC gamma-1. Both enzymes have pH domain and can thus bind the PIP3. These enzymes also contain SH2 domain that allow them to bind to the specific phosphorylated tyrosine residue on the LAP SLP76 complex, and it's here that ITK phosphorylate and activate PLC1. Activated PLC gamma-1 catalyze the hydrolysis of plasma membrane and phospholipid phosphatidylinositol 4,5 biphosphate, which is called PIP2, generating two breakdown product, the soluble sugar triphosphate inositol 1,4,5 triphosphate IP3 and membrane bound diacetylglycerol DAG. IP3 and DAG then activate two distinct downstream signaling pathways in T cell. IP3 produces a rapid increase in cytosolic free calcium after T cell activation. IP3 diffuses through the cytosol to the smooth endoplasmic reticulum, where it binds to its receptor, a ligand-gated calcium channel, and stimulates release of membrane sequestered calcium store. As a result of cytosolic free calcium, ion concentration increases from resting level about 100 nanometer to peak of 600 to 1000 nanometer within few minutes. The depletion of ER calcium is sensed by STIM1 and ER membrane protein, which then activate a plasma membrane ion channel called calcium release activated calcium CRAC channel. The portion of endoplasmic reticulum membrane in the cell are often very close to the plasma membrane, and this proximity facilitates the interaction between STIM1 and the CRAC channel. The activation of this CRAC channel results in an influx of extracellular calcium that sustain a cytosolic level at about 300 to 400 nanometer for more than 1 hour. A key component of crack channel is the ORI protein. Mutation in the gene encoding this protein are cause of rare human immunodeficiency disease. Cytoplasmic protein, cytoplasmic free calcium act as signaling molecule by binding to calmodulin, a ubiquin calcium-dependent regulator protein. Calcium calmodulin complexes activate several enzymes including calcineurin and protein serine threonine phosphatase that is important for transcription factor activation as discussed later. DAG, the second breakdown product of PIP2, is a membrane-bound lipid that activates the enzyme protein kinase. There are several isoforms of PKC that participate in generation of active transcription factor, discussed later. A combination of elevated free cytosol calcium and DAG induces conformation change in certain isoform of PKC that makes this catalytic site accessible to its membrane. Numerous downstream proteins are phosphorylated by PKC. The PKC later isoform localized to immune synapse, and it is involved in the activation of nuclear translocation of the nuclear factor. Nuclear translocation of the nuclear factor, kappa beta, transcription factor pathway. The NFKB activation are discussed later in this chapter. So far, we have described several signal transduction pathway initiated by ligand binding to the T-cell receptor that result in activation of different type of enzyme. The G protein map kinase pathway leading to activation of kinase such as ERK and JNK, a PLC gamma 1 calcium dependent pathway leading to activation of phosphatase calcineurin, and DAG-dependent pathway leading to activation of PK. Each of these pathways contribute to the expression of gene encoding protein needed for T-cell clone expansion, differentiation, and effector function. In the following section, we will describe the mechanism by which these different signal pathways stimulate the transcription of various gene T-cell. Activation of transcription factor that regulate the T-cell gene expression. The enzyme generated by T-cell receptor signaling activate transcription factor that binds to the regulatory region of numerous genes in T-cell and thereby enhance transcription of this gene. Much of our understanding of transcription regulation of gene in T-cell is based on an analysis of cytokine gene expression. The transcription regulation of most cytokine gene in T-cell is controlled by binding of transcription factor to the nucleotide sequence in the promoter and enhance region of these genes. For instance, the promoter located at 5' of the coding exons of the interleukin 2 genes contain a segment of approximately 300 base pair that contain binding site for several different transcription factor. All of these sites must be occupied by transcription factor for a maximum expression of interleukin 2 genes. Different transcription factor are activated by different cytoplasmic signal transduction pathway, and the requirements for the multiple transcription factor account for the need to activate many signal pathways after antigen recognition. The name the same principle are true for the induced expression of many other genes in T cell, including those encoding cytokine receptor and effector molecule, although different genes may be responsible for different combination of transcription factor. 3 transcription factors that are activated in T cell by antigen recognition and appear to be critical for the most T cell response are nuclear factor of activated T cell, AP1 and NF beta. NFAT is transcription factor required for expression of gene encoding interleukin 2, interleukin 4, and tumor necrosis factor and other cytokines. NFAT is present in active serine phosphorylated form in cytoplasm for resting T lymphocyte. It is activated by calcium calmodulin dependent phosphatase calcineurin. Calcineurin dephosphorylates cytoplasmic NFAT, thereby uncovering the nuclear localization signal that permit the NFAT to translocate in the nuclear. Once it is the nuclear, NFAT binds to regulatory region of interleukin-2 and other genes, usually in the association with other transcription factors such as UP1. The mechanism of activation of NFAT was discovered indirectly by studies of mechanism of action of immunosuppressive drug cyclosporine. This drug and functional similar compound tacrolimus are natural product of fungi that are used to as therapeutic agent to treat transplant rejection. They function largely by blocking T cell kinase by T cell cytokines gene transcription. Cyclosporin bind to the cytosolic protein called cyclophilin, and the tacrolimus bind to the protein called FK56 binding proteins, FKBP. Cyclosporin, cyclophilin complex, and the tacrolimus FKB complex bind to inhibit calcineurin. Hence, these drugs are called the calcineurin inhibitor and thereby block the translocation of NF into the nucleus. The UP1 is the transcription factor bond found in many cell types. It is specifically activated in T lymphocyte by T cell receptor mediated signal. UP is actually the name for a family of DNA binding factor composed of dimer of two proteins that bind to one another through a shared structural motif called leucine zipper. The best characterized UP factor is composed of the proteins FOS and JON. As discussed previously, the formation of active UP typically involves synthesis of the FOS protein and phosphorylation of pre-existing JON protein, both stimulated by MAP kinase that are activated by T cell receptor induced signal. UP physically associated with the other transcription factor in the nucleus, and it works best in combination of NAFT. Thus, UP activation represents a convergence point of several T cell receptor initiated signaling pathway. The NF-K beta refer to a group of closely related transcription factors that are activated in response to T cell receptor signal and are essential for cytokine synthesis. The NF-K beta protein are homodimer or heterodimer proteins that are homologous to serial and are important in transcription of many genes in diverse cell types. NF-K beta pathway is important not only for antigen receptor mediated lymphocyte activation, but also for response to signaling by innate pattern recognition receptors such as toll-like receptor and cytokine receptor, and is discussed in deep at the end of the chapter. These are structures that you can read. Get time, you have the book, read. And the link between different signaling protein, activation, transcription factor, and functional receptor response of T cell are difficult to establish because they are complex and incomplete understood interaction between signaling pathway. In addition, for the sake of simplicity, we often discuss signaling as set of linear pathway, but we know this does not reflect a more complex and interconnected reality. Finally, we have focused on selected pathway to illustrate how antigen recognition may lead to biochemical alteration, but it is clear that many other signaling molecules are also involved in antigen-induced lymphocyte activation. An additional mechanism by which T cell activation is regulated involves microRNAs, which are responsible for post-transcriptional inhibition of gene expression. Make are small non-coding RNA that are transcribed from DNA but not translated into protein. They are initially generated in the nucleus as longer protein transcripts that are processed by an endoribonuclease called drosha into shorter pre-microRNA that have a stem loop structure and can be exported into the cytosol. In cytosol, the pre-micro are processed by another ribonuclease called dicer into short double-stranded microRNA 21-22 base pair and lengths, which are associated with several proteins, including argonaute, to form complex known as RNA-induced silencing complex RISC. One strand of the micro can pair with a complementary sequence in number of cellular messenger RNA. If the 60 to 80 base pair micro sequence that recognize a messenger RNA is perfectly complementary to messenger RNA, the messenger RNA may be targeted for degradation, but if complementary is imperfect, translation of messenger RNA is inhibited. In either case, the result is the reduction in abundance of protein encoded by gene targeted by MYC or microRNA. In T cells, the expression of the majority of MYC is globally reduced upon activation. In addition, the argonaute protein is ubiquinated and degraded further, compromising the MYC RNA function and enhancing the expression of large number of protein required for cell cycle progression downstream of T cell activation. Modulation of T cell signaling by protein tyrosine phosphate. Tyrosine phosphatase remove phosphate moiety from residue on the protein and generally inhibit T cell signaling. Two tyrosine phosphatases that serve an important inhibitory roles in lymphocyte and other hematopoietic cell are called SHP1, SHP2. For SH2 domain containing phosphatase 1 and 2. Inhibitory phosphatases are typically recruited in items in the cytoplasmic tail of inhibitory receptor that are themselves phosphorylated for tyrosine kinase induced during lymphocyte activation. These phosphatases inhibit signal transduction by removing phosphate moiety from the tyrosine residue is key signaling molecule and thus functionally antagonize tyrosine kinase. Another inhibitory phosphatase called SHP SH2 containing SH2 domain containing inositol phosphatase does not act on phosphoprotein but rather in specific of inositol phospholipid. Like SHP1 and SHP2, SHP bind to phosphorylated item sequence on the specific inhibitory receptor. A SHP removes the phosphate group from PIP in the inner leaflet of the plasma membrane and thus antagonize the PI3 kinase signaling. Although most phosphatases attenuate lymphocyte signaling, one tyrosine phosphate, CD45, facilitates lymphocyte activation. The CD45 protein is a receptor tyrosine phosphatase expressed in all hematopoietic cells. It is an integral membrane protein whose cytoplasmic tail contains tandem protein tyrosine phosphatase domain. CD45 dephosphorate inhibitory tyrosine residue in SRC family kinase, including LCK and FYN in T cell, thus contributing to the generation of active kinase. Costimulatory receptor signal in T cell. Costimulatory signal are generated by a receptor that recognize ligand on antigen presenting cell and cooperate with the T cell receptor signal to promote activation of T cell. Two signal hypotheses for T cell activation was introduced in chapter 1 and chapter 4. In immunological jargon, the response by T cell receptor to MHC and peptide on an antigen presenting cell is referred to as a signal 1. T cells are fully activated only when a foreign peptide bound to the MHC molecule is recognized in context of activation of the innate immune system by a pathogen or some other cause of inflammation. Costimulatory reagent represent a danger signal, signal 2, induced by antigen presenting cell by microbes. Thus, recognition of a foreign antigen must be combined with sense of danger for optimal T cell activation. The CD28 family, of costimulatory receptor. The best defined costimulatory for T lymphocyte are a pair of related protein called B71, which is CD8, and B72, which is CD86, which are expressed on activated dendritic cell and other antigen presenting cell and bind to the CD28 receptor on T cell. CD28 molecule is a principal costimulatory receptor for delivery of secondary signal for T cell activation. The biologic role of B7 and CD28 protein families are discussed in chapters 9. Another activating receptor of CD28 family is a molecule called inducible costimulator, ICOS. Another activating factor receptor of the CD28 family is a molecule called inducible costimulator, ICOS, which play an important role in T follicular helper cell development and will be discussed in chapter 12. The CD2 or SLAM family of costimulatory receptor. Protein other than CD8 family member also contribute to T cell activation and differentiation. The CD2 family consists of homologous protein that play a role in activation of T cell and NK cell. In human T cell, CD2 function both as intercellular adhesion molecule and as signal transducer. A distinct subgroup of CD2 family of protein is known as SLAM, signaling lymphocyte activation molecule. Family. SLAM, like all members of CD2 family, is an integral membrane protein that contains two extracellular Ig domain and long cytoplasmic tail. The cytoplasmic tail of SLAM, but not of CD2, contain a switch motif that is distinct from item and atom motif found in other activating and inhibitory response. The extracellular Ig domains of SLAM are involved in homophilic interaction. SLAM on T cell can interact with SLAM on dendritic cell, and as a result, the cytoplasmic tail of SLAM may deliver signal to T cell. The items motif bind to the SAP SLAM associated protein and later form the bridge between SLAM and FYN, a SRI family kinase that is also physically linked to the CD3 protein in T cell. SLAM and other members of the SLAM family function as costimulatory receptor in T cell, natural killer cell, and some B cell. As we will discuss in chapter 21, mutation in H2DA gene encoding SAP are causes of disease called X-linked lymphoproliferative syndrome, XLP. An important member of SLAM family in natural killer cell, CD8 T cell, and gamma delta T cell is called 2B4. Like SLAM, the lymphocyte tail of 2B4 contain items bind to the SAP adapter protein and induce activating signaling by recruiting FYN. Defective 2B4 signaling contribute to immune deficiency in patients with X-linked lymphoproliferative syndrome. Metabolic change during T cell activation. When cells are activated, they need to increase their metabolic activity to cope with increased demand of cellular response. In immune system, this phenomenon has been best studied in T cell. Upon activation by antigen costimulatory, T cell increases transport of glucose and change their energy production from mitochondrial oxidative phosphorylation to glycolysis, even in the presence of abundant oxygen, a phenomenon known as aerobic glycolysis or Warburg effect. This was the first described in tumor cell but is now recognized as an important mechanism used by many proliferating cells. Although glycolysis generally less ATP, the molecule cell used a store to store and release energy than oxidative phosphorylation. Glycolysis does not use substrate other than glucose, such as amino acid and lipid, and thus preserve essential building block needed for synthesis of new macromolecule and for cell division. Aerobic glycolysis is in lymphocyte may be important not just for cellular proliferation but also for differentiation of T cell into effector cell and for production of effector cytokines. The B lymphocyte antigen receptor complex. So before we go for B cell, we see this is the T cell. Resting T cells normal. When it's now working, it changes from the oxidative phosphorylation to aerobic glycolysis, whereby it makes sure that it is generated less energy, but there's no utilization of amino acid protein. They are stored for it to proliferate, to increase in number, and to help multiplication of cell and function of the cell. So metabolic change during T cell activation. In resting T cell, the major pathway for energy generation in mitochondrial oxidative phosphorylation. Upon activation, there is a switch to aerobic glycolysis, which generates less energy but preserve and produce the building block for cellular organelle by biosynthesis, which is required for cell proliferation and functional response, ATP adenosine triphosphate. This one is B cell antigen receptor complex seen in left. This one and is associated with the invariant Ig beta and Ig molecule which contain items for cytoplasmic tails. Let us go and read about B cell. The B lymphocyte antigen receptor complex. The B lymphocyte antigen receptor is a transmembrane form of antibody molecule associated with two signaling chain. We describe the structure of antibody in detail in chapter 5. Here we will discuss some silence features of membrane form of Ig and the associated protein and discuss how they deliver signal to B cell. Because the signal pathway are much like those in T cell, we will summarize these without great detail. As noted earlier, there are some both similarity and significant difference between B and T cell antigen receptor, as we saw in chapter in table 7.1. The structure of B cell receptor for antigen. Membrane IgM and IgD, the antigen receptor of naive B cell, have short cytoplasmic tail consisting of only three amino acids: lysine, valine, and lysine. These tails are too small to transduce signal generated after the recognition of antigen. Ig mediated signal are delivered by two other molecules called Ig alpha and Ig beta that are disulfide linked to one another and are expressed in B cell, non-covalent associated with membrane Ig. These proteins have a function in B cell that are similar to the function of CD3 and Zeta proteins in T cell receptor signaling. They contain items in their cytoplasmic tail and are required for transport of membrane Ig molecule to the cell surface, and together with membrane Ig form the B cell receptor complex. The tail of Ig alpha and beta, Ig beta, are physically associated with the SRC family tyrosine kinase, including line, fine, and BLK. The B cell receptor complex is in class switching B cell, including memory B cell, contain membrane Ig that may be of IgG, IgA, or Ig class. See chapter 12. Membrane IgG and Ig molecule have longer cytoplasmic tail that contain a conserved aspartate or glutamate tyrosine adenine asparated methionine sequence called an Ig tail tyrosine motif that is similar in sequence and function to a signaling motif in the cytoplasmic tail of CD28 costimulatory receptor in T cell. Signal initiation by B cell receptor. Signal initiation by antigen occur by cross-link of the B cell receptor. Cross-linking of membrane IgM and IgD by multivalent antigen binding molecule Ig alpha and Ig beta associated SRC family kinase such as allele close to one another. Subsequent physical interaction of the kinase molecule activate this enzyme, enabling them to phosphorate the tyrosine residue and residue on the items of Ig alpha and Ig beta. The phosphorylation of item tyrosine residue trigger all subsequent signaling events downstream of the B cell receptor. A cross-link Ig receptor interlipid raft where many adapter protein and signaling molecule are concentrated. Along with SRC family, tyrosine kinase such as lin, fine, and BLK. The phosphorylated tyrosine residue in the item of Ig alpha and Ig beta provide docking site for the tandem SH2 domain of SYK tyrosine kinase. SYK is the homologous to ZAP2 and has a family function in B cell as ZAP70 and has similar function in B cell as ZAP70 does in T cell. SYK is activated, then is associated with phosphorylated kinase of items and may itself be phosphorylated on specific tyrosine residue by B cell receptor associated SRC family kinase, leading to the furthermore activation. Both SRC family kinase and SYK contribute to the activation of BTK and an important tyrosine kinase in B cell. If the antigen is monovalent and incapable of cross-linking multiple Ig molecule, some signaling may nevertheless occur, but the additional activation by helper T cell may be necessary to fully activate the B cell, as discussed in chapter 12. So let us see. Cross-link of membrane Ig by antigen. You see this is multivalent antigen. These are the receptors, so there is a cross link. This helps a lot in a stimulation of B cell because it has less and less receptors. So here we are told single transduction by B cell receptor complex antigen induces cross link of membrane of immunoglobulin Ig on B cell lead to the clustering and activation of SRC family tyrosine kinase and tyrosine phosphorylation of the items in the cytoplasmic tail of the Ig alpha and Ig beta molecules. This leads to the docking of SYK and subsequently tyrosine phosphorylation event as depicted. Several signaling cascades follow this event as shown, leading to the activation of several transmembrane transcription factor. These signal transduction pathway are similar to those described in T cell, AP activation, activation protein, gene guanosine diphosphate, GPP guanosine triphosphate items, and immunoreceptor tyrosine based activation. Motif, NFAT nuclear factor of activated T cell, NFKB nuclear factor, NKB. PLCY is a phosphate C gamma 1. P is phosphatidyl triphosphate. Yeah, those are the PKC is protein kinase C. So, you see the structure tyrosine phosphorylation activating the CRC kinase, the family, this one is Leon, FAN, and BLK. It is activated, this one phosphorylation will happen. A lot of things will happen here. You can follow them, and biochemical intermediate to get the PLC gamma activation together with GTP, GDP exchange on RAS and RAS. Then this one causes inositol triphosphate increase in cytoplasmic calcium, diphosphate DAG increases. Then you have active enzymes. Then transcription factor of MAC, NAFT, and other things will happen, go to the cytoplasm. Then they go they will go to the nuclear for other function. Role of CR2CD21 complement receptor as co-receptor for B cell. The activation of B cell is enhanced by signals that are provided by complement protein and the CD21 co-receptor complex, which link in innate immunity to the adaptive humoral immune response. Microbial surface and release the polysaccharide can activate the complement system by alternative and lectin pathway during innate immune response in the absence of antibodies. See chapter 4, chapter 14. Protein and other antigens that do not activate complementary directly may be bound to pre-existing antibody or by antibody produced earlier in response, and these antigen-antibody complex can activate complement by a classical pathway. Recall that complement activation results in proteolytic cleavage of the complement protein. The key component of system is a protein called C3, and its cleavage results to the production of molecule called C3b that bind covalently to the microbes or antibody complex. CB may be further degraded into the fragment of called C3d, which remain bound to the microbial surface on the antigen complex. The B lymphocyte expresses a receptor for C3d called the type 2 complement receptor, CR2 or CD21. The complex of C3d and antigen or C3d and antigen antibody complex bind to the B cell, with the membrane Ig recognizing antigen and CR2 recognizing the bond to C3d. So this is complement. We have the microbes, micro bond to C3d. You see this bond C3d. Then we have recognition of B cell. This is IgM and this is CR2. This is like complement. Then when it is binded by both, what happens is there is a very good activation of the B cell. So role of complement in B cell activation. B cell expresses a complex of CR2 complement receptor, CD19 and CD81 microbial antigen that have bound the complement fragment C3d can simultaneously engage both the CRT molecule and membrane immunoglobulin Ig on surface of B cell. This leads to initiation of signaling cascade from both B cell receptor complex and CR complex, because of which the response of C3d antigen complexes is greatly enhanced compared with the response to antigen alone. So, CR2 is expressed on mature B cell as complex with two other membrane protein, the CD19 and CD81, also called the TAPA1. The CR2 and CD19 CD81 complex is often called the B cell co-receptor complex because CR2 binds to the antigen through attached C3d at the same time that membrane Ig binds directly to the antigen. Binding of C3d to the B cell complement receptor brings CD19 in proximity to B cell receptor associated kinase, and cytoplasmic tail of CD19 rapidly become tyrosine phosphorylated. This leads to the activation of PI3 kinase, which generate PIP3, which in turn bind to the activate BKT and PLCY gamma 2 in the manner analogous to the PDK1 activation and PLC gamma 1 activation in T cell. The net result of co-receptor activation is that the response of antigen-stimulated B cell is greatly enhanced. Complement protein are not the only innate immune trigger of B cell. Other innate immune ligands, including the flagellin and microbial nucleic acid, can also activate B cell via Toll-like receptor. Toll-like receptor signaling has been discussed in chapter 4, and the role in activation of B cell will be considered in chapter 12. Signal pathway downstream of B cell receptor. After antigen binding to B cell receptor, SYK, the other tyrosine kinase activate numerous downstream signaling pathways that are regulated by adapter protein. Activated YK phosphorylate the critical tyrosine residue on adapter proteins such as SLP65, also called the BLNK. This facilitates the recruitment of these adapter proteins of the SH2 domain and phosphotyrosine binding PTB domain containing enzyme including guanine nucleotide exchange protein that can separately activate RAS and RAC, PLC, gamma 2, and BTK tyrosine kinase, among others. Recruitment facilitate activation of these downstream effectors, each generally contributing to the activation of distinct signaling pathway. The RAS-MAP kinase pathway is activated in antigen-stimulated B cell. The GTP-GDP exchange factor source is recruited to adapter protein SLP65 through the binding of GRB2. The RAS is then converted by SOC from the inactive GDP bond form and active GTP bond form. Activated RAS contribute to the activation of ERK-MAP kinase pathway as discussed earlier in T cell signaling. In parallel fashion, the activation of small G protein RAS may contribute to the activation of JNK-MAP kinase pathway. A phosphatidyl inositol specific phosphatase C, PLC, is activated in response to BRC signaling, and this in turn facilitates the activation of downstream signaling pathway. In B cell, the dominant isoform of PLCY is the alpha 2 isoform, where T cell expresses a related gamma 1 isoform of enzyme. PLC gamma 2 become active when it binds to the adapter protein BLNK and is phosphorylated by BTK. As described in the context of toll-like receptor signaling, active PLC break down membrane PIP2 to yield a soluble IP3 and leaves tags in the plasma membrane. IP3 mobilizes calcium from intracellular store, leading to the rapid elevation of the concentration of cytoplasmic calcium ion, which is subsequently augmented by influx of calcium from extracellular milling. Calcium signaling contributes to the NFAT activation in B cell as well, analogous to event described in T cell. In the presence of elevated calcium, DAG activates some isoform of PKC, mainly PKCB beta, in B cell, which phosphorylate downstream protein on the serine threonine residue. PKCB activation downstream of B cell contribute to the activation of NFKB antigen stimulate B cell. This process is similar to that in T cell triggered by PKCO, the PKC isoform present in T cell. As described for T cell activation, phosphorylation of a specific tyrosine containing motif on a number of adapter in B cell allow the recruitment of activation of PI3 kinase. This enzyme facilitates critical cellular events, including cell survival in activated B cell. This signaling cascade ultimately leads to the activation of transcription factor that induce the expression of genes whose products are required for functional response of B cell. Some of the transcription factors that are activated by antigen receptor mediated signal transduction in B cell are the downstream of RAS and ERK activation, JAN downstream of RAC and JNK activation, and NFK beta downstream of BTK, PLC gamma 2, and PKC beta activation. We described this earlier in the context of T cell signaling pathway. These and other transcription factors not mentioned here are involved in stimulating proliferation and differentiation of B cells in chapter 12. Same signaling pathway are used by membrane IgM and IgD on naive B cell, and by IgG, IgA, and IgE on B cell that have undergone isotope switching because of all of these membrane Ig isotope are associated with Ig alpha and beta. However, in memory B cell expressing membrane IgG and IgE, the tyrosine residue in the ITT motif phosphorate recruit the GRB2 adapter and enhance both ERK activation and calcium signaling. The attenuation of immune receptor signaling. Activation of lymphocyte has to be tightly controlled to avoid collateral damage to host tissue during immune response against foreign antigen. In addition, the immune system needs mechanisms that prevent reaction against self-antigen. We describe the biology of these control mechanisms in later chapters, mainly chapter 15. Here we discuss some of the biochemical mechanisms that limit the and terminate lymphocyte activation. Inhibitory signaling in lymphocyte is mediated primarily by inhibitory receptor and also by enzyme known as E3 ubiquin ligase that mark certain signaling molecule for degradation. Functional response of all cells are regulated by a balance between stimulated and inhibitory signal. Many inhibitory receptors recruit and activate phosphatase that counter kinase dependent signaling event induced by receptor for antigen and costimulators. We will describe how inhibitory receptors may function in natural killer cell and T cell and B cell, and then now ubiquin 3 ligase may attenuate signaling in lymphocyte. The biological relevance of signal attenuation through inhibitory receptor in natural killer cell, T cell, and B cell is addressed in chapter 4 for 15 and 12 respectively. So we have the inhibitories, ligands, the items, SRI family kinase, all these. Inhibitory signal in lymphocyte. A schematic depiction is provided of an inhibitory receptor with extracellular ligand binding domain and cytosolic immunoreceptor tyrosine based inhibition motif, ligand binding results. And in phosphorylation of item tyrose by SRC family kinase, followed by recruitment of SH2 domain containing tyrosine phosphatase that can remove phosphatase from signaling intermediate and thus attenuate immune receptor signaling. Inhibitory receptor of natural killer cell, B cell, and T cell. Inhibitory receptor plays a key role in natural killer cell, T cell, and B cell, and in other cells of innate immunity. Most but not all inhibitory receptors in the immune system contain items in their cytoplasmic tail that can recruit SH2 domain containing phosphatase and thus attenuate signaling in broadly similar manner. Tyrosine residue in items of this receptor can phosphorylated by SRC family kinase linked to lymphocyte activation and, as described earlier, recruit SH2 domain containing tyrosine phosphatase such as SHP1, SHP2, and SH2 domain containing isotyl phosphatase SHIP. SHP1, SHP2 attenuate tyrosine kinase initiated signaling from activating receptor in natural killer cell and from the B cell receptor and T cell receptor in B cell and T cell respectively. A SHIP remove phosphatase moieties from PIP3 as described earlier and thus inhibiting the P13 kinase activity in lymphocyte, natural killer cell, and innate immune cells. The natural killer cells inhibitory receptor called killer cell immunoglobulin receptor, KIR, seen chapter 4, contain extracellular Ig domains that can recognize class 1 human leukocyte antigen HLA molecule and subset of this receptor contain cytosol. Items: the CD94 NKG2A inhibitory receptor binding to a typical class 1 MHC molecule called HLIE, and NKG2A chain of this dimer contains cytosolic items. The major inhibitory receptor of T cell are proteins of CD28 family. They are also called the co-inhibitor to contrast them with the co-stimulators. One of these is CTLA for CD152, has higher affinity than CD28 for B7 protein and is competitive inhibitor of B7 CD28 interaction. It inhibits immune response mainly by outcompeting the activating receptor. CD28 for its ligands, the B7 costimulator, and by removing B7 molecule from antigen-presenting cell. It is involved in maintenance of unresponsiveness tolerance to the self-antigen and is discussed in this context in chapter 15. Another co-inhibitory receptor of the same family is PD-1. This is also discussed in chapter 15. PD-1 contains cytosolic items and items that both contribute to the inhibitory signaling in T cell. The items, when phosphorylated, recruit a tyrosine phosphatase, SHP2, which block T cell activation mediated by T cell receptor complex and CD28. Fc gamma IIB is an important attenuator of signaling activated B cell, dendritic cell, and macrophage, and it can bind to IgG containing immune complex through extracellular Ig domain and primarily recruit SHP and antagonize PI3 kinase signaling. This receptor dampens B cell activation after antibodies are produced and will be discussed in more detail in chapter 12. Ubiquin-dependent degradation signaling protein. One of the major ways for degrading cytosolic and nuclear protein involve the covalent attachment of ubiquin residue to this protein. Although ubiquination of protein is frequently linked to the degradation of this protein in proteasomes, proteins can be ubiquinated in a number of ways, each form of ubiquination serving a different function. In context of signal transduction, two distinct types of ubiquination mediate signal attenuation on the one hand and signal generation on the other hand. Ubiquination was briefly discussed in chapter 6 in the context of the class 1 MHC pathway of antigen processing. Ubiquination is a 76 amino acid protein that is activated in ITP-dependent fashion by E1 enzyme, then it transferred to E2 enzymes, which then covalently attach the activated ubiquin to lysine residue on the specific substrates that are recognized by specific E3 ubiquin ligase. In many cases, after C-terminus of ubiquination moiety covalently linked to lysine residue on the target protein, the C-terminal ends subsequent ubiquination moieties may be covalently attached to lysine residue on preceding ubiquin to generate polyubiquin chain. The shape of polyubiquin chain is different depending on which specific lysine residue on the preceding ubiquin molecule in the chain is the site of covalent binding of the next ubiquin molecules, and the shape of ubiquin chain has important function. Consequence: if lysine in position 48, the first ubiquin moiety form an isopeptide bond with a C-terminus of the next ubiquin, and so on. Lysine 48 type of ubiquin chain will be generated that can be recognized by proteasome cap, and ubiquin protein will be targeted for degradation in a proteasome. Some E3 ligases generate different types of polyubiquin chain, which does not target proteins for degradation but instead generates a structure for latching the marked protein and other specific protein. This is important in NFKB K-bet signaling as discussed later. For some function, in particular targeting membrane protein to lysosome rather than to protosome, only single ubiquin moiety may need to be attached to protein target. Several E3 ligands are found in T cell, some of which are involved in signal activation and other signal attenuation. The prototypes of E3 ligands involved in terminating T cell response is CBL, B, but several other serve similar function. Recruitment of CBLB to the T cell receptor complex and associated adapter protein lead to the monoubiquination, endocytosis, and lysosome degradation of T cell receptor complex, and this may be a mechanism for attenuation of T cell signaling. CD28 signal block the inhibitory activity of CBLB, and this is one of mechanisms by which costimulatory augment the T cell signaling. In knockout mice lacking the CBLB, the T cell respond to antigen even without CD28 mediated costimulatory and produce abnormal high amount of interleukin 2. These mice develop autoimmune as a result of excessive activation of the T cell. There is some evidence that antigen that shut off immune response called the tolerogenic antigen, such as self-antigen, activate in T cell ubiquin ligands that degrade essential signaling proteins, and this is mechanism of antigen-induced unresponsiveness called anergy. See chapter 15. Let us learn about cytokines, receptor, and signaling. Cytokine, the secreted messenger molecule of immune system, have been mentioned in previous chapter, and will be throughout the book. Here we will describe the receptor for cytokine and their mechanism of signaling. All cytokine receptor consists of one or more transmembrane proteins whose extracellular portion are responsible for cytokine binding, and those cytoplasmic portion are responsible for initiation of intracellular signaling pathway. For most cytokine receptor, these signaling pathways are activated by ligand-induced receptor clustering, bringing together the cytoplasmic portion of the two or more receptor molecule and thus inducing the activity of unique non-receptor tyrosine kinase. In case of tumor necrosis factor receptor family of the cytokines receptor, preformed receptor trimer undergo a conformational change after contacting their cognate trimetic ligands. So, role of ubiquin ligands, we studied it later back in the past chapter. So we see the role of ubiquin later. We have already discussed. Now we are continuing cytokines. The classes of cytokines receptor. The most widely used classification of cytokine receptor is based on the structural homology of extracellular cytokine binding domains and shared intracellular signaling mechanism. Signaling mechanisms used by individual family are considered in the section that follows. The tumor growth factor, the transforming growth factor beta family is one of the widely studied family of growth factor not included in the classification figure 7.23, and its signaling mechanism discussed later. Type 1 cytokine receptor, hematopoietin receptor family. Type 1 receptor are dimer or trimer that typically consist of unique ligands binding chain and one or more signal transducing chain. The latter often shared by receptor for different cytokines. These chains contain one or two domains with a conserved pair of sustained residue and a membrane proximal peptide stretch containing a tryptophan, serine alpha, tryptophan serine. Motif, where X is any amino acid. The conserved sequence of the receptor form a structure that bind cytokines that have 4 alpha helical bundle and are referred to as type 1 cytokine, but the specificity for individual cytokine is determined by amino acid residue that vary from one receptor to another. This receptor family can be divided into subsequent based on structural homologism or the use of shared signaling peptide. One group, which includes receptor interleukin 2, interleukin 4, interleukin 7, interleukin 9, interleukin 15, interleukin 21, containing signaling component called common gamma chain, gamma C and CD132. Within this group, some receptors share one or two beta chains subunity, CD122, CD131, or CD131, and some lack beta chain. Another subgroup of type 1 cytokines receptor, including those of interleukin 6, interleukin 11, and interleukin 27, use the GP130 signaling chain. All the type 1 cytokine receptor engage in JAK-STAT signaling pathway, as discussed later. Type 2 cytokine receptor interferon receptor family. The type 2 receptor are similar to type 1 receptor by virtue possessing two extracellular domain with conserved cytosine, but type 2 receptor do not contain the YCXY WX WX WSXYS motif. All of the type 2 cytokine receptor, like the type 1 receptor, engage in JAK-STAT signaling pathway. This family include receptor of interferons, that is interferons, and interleukin 10, interleukin 20, and interleukin 22. Tumor necrosis factor receptor family. These receptors are part of the large family often called TNFR superfamily of preformed trimer, some of which recognize membrane-associated ligand and are not considered cytokine receptor, with the conserved cysteine-rich extracellular domain and shared intracellular signaling mechanism that typically stimulate gene expression but in some cases induce apoptosis. The important receptor for this family will be discussed on other chapters in their biology context. They include tumor necrosis factor receptor, tumor necrosis factor receptor 1, tumor necrosis factor receptor 2, the CD4 proteins, FAS, the lymphotoxin receptor, and the BAF receptor family, among others. The ligands for these receptors also form trimers. Some of these ligands are membrane bound and other are secreted soluble protein. So here we are having cytokine receptor families. We have type 1 cytokine, which are mostly found in hematopoietin receptor. These are conserved cytokines, ligands, the interleukins 2, 3, 4, interleukins 5, 6, 7, 9, 11, 12, 13, 15, and all others here together with this one. Then type 2 cytokine receptor, these are most of them they are interferons, interleukin also, interleukin 10, 20, 22, interleukin 24, interleukin 26. The tumor necrosis factor receptor subfamily, we see them, the BAF, the CD40 ligands, the FALS, the LEAS, and interleukin 1 receptor family. These are the interleukin 1, interleukin 18, interleukin 33. We have interleukin 17 receptor family, interleukin 17A, interleukin B, C, D, interleukin E, interleukin 27, and we have chemokines. The structure of cytokine receptor A receptor for different kinds are classified into family on basis of conserved extracellular appearance, domain and structure and signal mechanism, representative cytokines and other ligands that bind to each receptor family are listed below for schematic drawing. Big group of cytokine receptors share identical or highly homologous subunit chain. Selected example of cytokines receptor in each group are shown. In common gamma chain family, the interleukin 2, interleukin 15 receptor share beta chain CD122. In the common beta chain family, shared beta chain is CD130. Granulocyte colon stimulating factor GM-CSF, granulocyte macrophage colon stimulating factor, interleukin, tumor necrosis factors, and etc. By the binding of the ligand to preformed trimetic receptor typically induce a conformational change and recruit adapter protein to receptor complex. These adapter in turn recruit enzymes that include both E2 Ubiquin ligase, which mediate non-degradatory polyubiquitination, and protein kinase, which initiate downstream signaling. In case of the tumor necrosis factor receptor restricted, the receptor recruits the adapter protein tumor necrosis factor receptor associated death domain, and the trade in turn can recruit protein called TRAF receptor associated factor, which possesses a unique type of E3 ligase activity that will be discussed in the section of NF-kappa signaling. The type 1 TNF receptor, there are two different receptors for TNF and FAS CD95, can also recruit adapters that lead to the activation of caspase 8, and this receptor can thereby induce apoptosis in certain cells. So cross-linking of TNF, FR1, like tumor necrosis factors, they can cause death domain, activation effector caspase, lead to the apoptosis, things like that. So interleukin 1 family. The receptor of this family share a conserved cytosolic sequence called the toll interleukin 1 receptor domain and engage similar signal transduction pathway that induce new gene transcription. We discussed toll-like receptor signal in chapter 4. Briefly, engagement of interleukin 1 receptor or toll-like receptor results in receptor dimerization and recruitment of one or more of the four known TIR domain containing adapter to the TRI domain of the cytoplasmic tail of the receptor. The adapters link toll-like receptor to different members of interleukin 1 receptor associated kinase IRAC family. IRAC can in turn link adapter to TRAF6 and AR3 ubiquin ligase required for NF-kappa beta activation. Other events downstream the toll-like receptor signal include MAP kinase activation and phosphorylation of IRAF3, IRAF7 induce of type 1 interferon gene transcription. The latest aspect of toll-like receptor induced toll-like signaling has been considered in the context of antiviral statin chapter 4. Different adapter link toll-like receptor to NF-kappa beta signaling, MAP kinase activation, and activation of IRF3. The mechanism connecting interleukin 1 receptor, toll-like receptor signaling, and NF-kappa beta activation are discussed later. Interleukin 17 family. The receptor of this family are preformed oligomers that include various combinations of interleukin 17R, A, B, C, D, and E chain. Receptor oligomers include at least one molecule of interleukin 17RA chain. Each receptor chain is a type 1 integral membrane protein that contains two extracellular type 3 fibronectin domain and an intracellular self motif, which has partial homologue to TRF motif discussed in the context of interleukin 1 receptor signaling. The self motif, however, does not recruit adapter that bind the to toll-like receptor, the interleukin-1 receptor. This motif binds to an adapter ACT1 that also contain a self motif and contribute to the recruitment of TRA and lead to the NF beta activation. Additional cytoplasmic motifs are required for activation of many other pathways, including the ERK pathway and the CEBK family transcription factor. So the cytokines structure we're going to see. There are many different cytokines of interleukin-17 family, and in subsequent chapter the emphasis will largely be on cytokines that are best characterized in the context of autoimmune and inflammation, namely interleukin-17A, interleukin-17F, interleukin-17B, C, and D remain poorly characterized, and interleukin-17A, also known as interleukin-25, drives Th response. Interleukin-17A homodimer, interleukin-17F homodimer engage preformed heterodimer made up of interleukin-17Ra and interleukin-17Rc. Interleukin-17E, interleukin-25 engage a dimer containing interleukin-17Rb and interleukin-17Ra. Signaling by JAK and STATs. Cytokine receptor of the type 1 and type 2 receptor family engage signal transduction pathway that involve non-receptor tyrosine kinase called Janus kinase, JAKs, and transcription factor called signal transducer and activation of transcription, STATs. The discovery of JAK-STAT pathway came from a biochemical and genetic analysis of interferon signaling. There are 4 known Janus kinase, JAKs1, JAK2, JAKs3, and TYK2, and 7 STAT, which are signal transducer and activators of transcription. 1 to 4, 5A, B, and 6. The sequence of events in JAK-STAT signaling pathway is now well defined. Inactive JAK enzymes are non-covalently attached to cytoplasmic domain of type 1 and type 2 cytokine receptor. When two receptor molecules are brought together by binding of a cytokine molecule, the receptor-associated JAK are activated and phosphorylate tyrosine residue in the cytoplasmic portion of clustered receptor. Some of these phosphotyrosine moieties of the receptor are then recognized by and bind SH2 domain of monomeric cytosol STAT protein. The STAT proteins are thus brought close to JAK and phosphorylated by this receptor-associated kinase. The SH2 domain of one STAT monomer is able to bind to phosphotyrosine residue on the adjacent STAT protein. The STAT dimer that are generated migrate to the nucleus where they bind to specific DNA sequence in promoter region of cytokine response genes and activate gene transcription. An intriguing question is how the specificity of response to many different cytokines is achieved. Given the limited number of JAK and STATs used by a large number of cytokine receptors, the likely answer is that unique amino acid sequence in different cytokine receptors provide a scaffold for specifically binding and thereby activating different combination of JAK and STATs. The SH2 domain of different STAT proteins selectively bind to phosphotyrosine and flanking residue of different cytokine receptor. This is largely responsible for the activation of particular STATs by various cytokines receptor and thereby for specificity of cytokines signaling. Several type 1 and type 2 cytokine receptor are heterodimer of two different polypeptide chain, each of which bind a different JAK. Furthermore, two different starts may heterodimerize on phosphorylation. Thus, there is a significant amount of combinatorial diversity in signaling that can be generated from a limited number of JAK start protein. The subset of type 1 cytokines receptor that uses the common gamma chain all uses the JAK3 kinase of signaling. JAK3 is the only JAK kinase that is not expressed ubiquitously. Its expression is largely restricted in immune cell, and it is only activated by gamma C containing receptor. Type 1 cytokine receptor of interleukin 6 family use JAK2 to activate STAT3. A number of other cytokines will also activate STAT3. Several JAKs and STAT are relevant to human disease and target therapeutic agents. Gain of functional mutation in JAK2 are the cause of myelodysplastic syndrome with aplastic anemia and polycythemia vera. Mutation affecting this gamma C chain, or less frequently, a JAK3 causes severe combined immunodeficiency, seen in chapter 21. Dominant negative mutation in STAT causes immunodeficiency disease due to defect in T helper 17 response. Activating mutation in STAT3 are characteristic features of the large granular lymphocyte leukemia that are malignant proliferation of cell of natural killer cell or the CD8 T cell lineage. The elucidation of JAK-STAT signaling has led to the development of novel therapeutic agents targeting this pathway. Small molecule JAK antagonists are approved for treatment of acute myeloid leukemia and some other myeloid malignancies, and also for certain chronic inflammatory diseases including rheumatoid arthritis and psoriasis. Cytokine activates signaling pathway and transcription factor in the activation in addition to the JAKs and STAT. For instance, the interleukin 2 receptor beta chain activates RAS-dependent MAP kinase pathway that may be involved in gene transcription growth stimulation. Other cytokine receptor may similarly activate other signaling pathway in consent with JAK-STAT pathway to elicit biological response to the cytokines. T proliferation triggered to be a considerable degree by cytokines such as interleukin 2 is targeted by some immunosuppressive small molecules. An important downstream protein kinase that regulates the protein translation and cell growth in many cell types, including dividing T cell in mTOR. It is, as its name implies, inhibited by rapamycin, a clinically used immunosuppressive drug. Several mechanisms of negative regulation of JAK-STAT pathway have been identified. Protein called suppressor of cytokine signaling SOX serve as adapter for multi-subunity E3 legs activity. They can bind to activated STATs and JAKs and the tightly associated E3 ligase ubiquinate, the JAK and STAT targeting them for proteasomal degradation. SOX protein can be degraded by toll-like receptor ligands, by cytokines themselves, and by other stimuli. In this way, SOX protein serves as negative feedback regulator of cytokines mediated activation of cell. Other inhibitors of JAK-STAT signaling include tyrosine phosphatase such as SHP1, SHP2, which can dephosphorate and therefore deactivate JAK molecule. Another family of inhibitory protein called protein inhibitor of activated STAT plus bind phosphorylated STAT and prevent their interaction with DNA. It is now known that PS protein also interact with and block the function of other transcription factor associated with cytokine signaling, including NFK beta and SMAD transcription factor downstream, the member of tumor growth factor beta receptor family. Pathway for NF kappa beta activation. NF kappa beta refer to a group of structural related transcription factor that play a central role in inflammation, lymphocyte activation, cell survival, and formation of secondary lymphoid organ. NF beta family member are important player in lymphocyte development and is in the pathogenesis of many cancers, including malignant neoplasm derived from activated lymphocytes. NF beta is promptly activated by cytokines of interleukin 1, tumor necrosis factor, interleukin 17 family entities, also induced downstream of toll, and is also induced downstream of toll-like receptor stimulation and antigen recognition. It is discussed here as prototype of transcription factor with fundamental roles in innate and adaptive immunity. There are five NF kappa beta protein. The domain that is common to all NF kappa protein is DNA binding domain called rel homologue domain. For transcription factor to be active, it must both bind to DNA and contain an activation domain that can facilitate transcriptional initiation. 3 NF beta protein have both rel homologue domain and activation domain. These are P65 rel A, rel B, and C rel. 2 protein FK beta 1 P50 NFK beta P52 contain DNA binding rel homologue domain but lack activation domain. NFK beta B1 typically form active heterodimer with P65 rel A or with C rel, and NFK beta form heterodimer with rel B. There are two pathways for NFK activation called conical and non-conical pathway. Most stimuli that initiate the NFK beta kappa do so by inducing the conical pathway, hence its name. This pathway is activated by a number of receptors that drive inflammation, such as toll-like receptors, interleukin 1 receptor, and some members of tumor necrosis factor receptor families, such as tumor necrosis factor receptor 1. It's also activated in context of lymphocyte activation by B cell receptor and T cell receptor. The conical pathway results in a nuclear localization of transcription active heterodimer of NFK beta with P65 rel A with C rel. NF kappa beta 1 containing heterodimer normally reside in the cytosol bond to an inhibitor of NFK beta called 1 kappa beta alpha, and they cannot access the nucleus in non-activated cell. The conical NFK pathway induces the tagging and degradation of I kappa B alpha, allowing the unaffected heterodimer NF kappa beta contained transcription factor to migrate into the nucleus. Two very different types of polyubiquination events are required for conical NF kappa beta activation. There are few common steps in the conical pathway that apply to all upstream signal input. Upstream signaling leads to the activation of unique type of ubiquin E3 ligase that can add the lysine lysine 63 type of ubiquin chain to protein called NEMO or IKK gamma, that is non-catalytic subunit of trimetric enzymes complex called I kappa beta kinase. This complex contains two other subunits called IKK alpha, IKK beta, both of which have a potential to be catalytically active serine threonine kinase. Ubiquitination of NEMO allow IKK beta to be activated by upstream kinase. Active IKK beta phosphorylate as inhibitory protein bound to NF-K beta I kappa beta alpha on two specific etheric residue and thus tag this protein for lysine 48 ubiquination. Polyubiquinated IK beta alpha is targeted by degradation in the proteasome and conical NF kappa beta heterodimer is then free to enter the nucleus. So this is the structure. Conical pathway, you have tumor necrosis factor family receptor, tumor necrosis factor. You have here what is happening, toll-like receptor, antigen receptor. So actually you can read it more and more and get more understanding. So we have discussed earlier how T cell receptor and B cell signaling contribute to the activation of PKCO and PKC beta respectively. This PKC can phosphorylate protein called CARMAR1 that forms a complex with two proteins called BCL10 and MALT1. The CARMAR1 MALT1 BCL10 complex can contribute to the activation of lysine 3 type of ubiquitin E ligase called TRF6. Active TRF6 can activate TAC and also add the lysine 6-3 ubiquitin chain to NEMO, thus facilitating the activation of IKK beta, toll-like receptor, interleukin 17R, and the interleukin 1R also activate the TRAF6 to initiate the IKK activation. Many members of tumor necrosis factor receptor family, including the tumor necrosis factor receptor and CD40, can activate conical NF kappa beta signaling through the activation of other proteins, the TRF3 and TRF5. A separate NF signaling pathway called the non-conical pathway processes a precursor protein called P100 into P52, thus allowing the heterodimer of NF kappa and its partner Relay to enter the nucleus. In a non-activated cell, the P3 precursor is bound to RelB and P100 RelB complex is unable to enter the nucleus until P100 is converted to P52. This pathway is activated downstream of few tumor necrosis factor receptor family signaling receptor, most notably the lymphotoxin beta receptor that drive the lymphocyte organogenesis and the B and BAF receptor that facilitate B cell survival. Receptors such as LT beta receptor and BAF that induce the non-conical NF kappa beta pathway also uses TARF to activate the kinase, a kinase called NIC, that in turn activates an IKK-like complex that contain IKK alpha homodimer. This leads to phosphorylation of P100, making it for ubiquitination and degradation in the cytosol, lead to the generation of non-conical P52 REL B NF kappa beta complex that can immigrate into nucleus. So, tumor growth transforming growth factor beta signaling. Transforming growth factors factor beta 1 is growth factor importance is the growth factor of importance in T and B cell biology. It is one member of the widely studied transforming growth factor beta superfamily of structure-related growth factor that play a crucial role in numerous aspects of in vertebrate and vertebrate development as well as in cellular transformation in cancer. During T cell differentiation, transforming growth factor beta participates in regulating T cell development and development. So it's regulate the T cell development. So during T cell differentiation, transforming growth factor participate in regulatory T cell development and development of T helpers 17 cells. In humoral immunity, transforming growth factor is important for class switching to IgA. Newly synthesized transforming growth factor beta is cleaved into Golgi in the Golgi into the fragmented C-terminal peptide that dimerizes to form inactive cytokine and an N-terminal pro-domain called latent-associated peptide that also dimerizes and remain bound to inactive tumor growth factor beta dimer. After secretion, the latent-associated peptide dimer still bound to inactive T cell tumor transforming growth factor beta dimer associated covalently, forming a disulfide bond with specific protein on the extracellular matrix, forming what is known as latent complex alpha V integrin on the another cell bind to the latent associated peptide, and the activated integrin then excites physical force, disrupting the latent complex and leading to the release of active T transforming growth factor beta dimer. Transforming growth factor dimer then bind to the dimetric transforming growth factor beta R11 receptor protein on adjacent cell surface. Mammalian cell contains 7 different tumor growth factor beta family type 1 receptor and 5 different types 2 receptor. Both transforming growth factor beta R1 and transforming growth factor beta R2 protein are transmembrane protein dimer whose extracellular domain bind to transforming growth factor beta and whose cytosolic tail contains serine threonine kinase domain. After transforming growth factor beta RR2 has been ligated, it is catalytically activated and phosphorylated and activated TG transforming growth factor beta R1. Catalytically active transforming growth factor R1 then phosphorylates the cytosolic receptors SMAD family transcription factor or RMSMAD, such as SMAD-D or SMAD-D3, which then require high affinity for co-SMAD such as SMAD. The R-SMAD and co-SMAD complex then goes to the nuclear induced transcription of tumor growth and the transforming growth factor beta target genes. Depending on cell type, these target cells differ. Induction of peripheral or induced regulatory T cell, for instance, seen chapters 15. Tumor growth factor beta signaling induce the expression of the FOX transcription factor, which is essential for developmental and inhibitory function of this regulatory T cell. In summary, it has been a very long topic, so let us read the summary. So, signaling receptor typically located on cell surface generally initiate signaling in the cytosol, followed by nuclear phase during which gene expression is altered. Very important point. Signaling receptor typically located on cell surface generally initiate signaling in cytosol, followed by nuclear phase during which gene expression is altered. Many different types of signaling receptor contribute to the innate and adaptive immunity, the most prominent category being in immune receptor that belong to the receptor family in which non-receptor tyrosine kinase phosphorylate tyrosine containing immunoreceptor tyrosine based activation item on the cytoplasmic tail protein in the receptor complex. Some of the types of receptor of interest in immunology include those of the receptor tyrosine kinase family, nuclear receptor, heterotrimetic G protein coupled septins receptor, and receptor of the Notch family. Antigen receptor on T cell, as well and B cell, as well as immunoglobulin Ig, Fc receptor, are member of immune receptor family. Antigen receptor can produce wide varying outputs depending on the affinity and valence of antigen that can recruit different number of items. Core receptors such as CD4 or CD8 on T cell and CD21 CR2 on B cell enhance signaling from antigen receptor. Co-receptor bind to same antigen and complex that is being recognized by antigen receptor. Signaling from antigen receptor can be attenuated by inhibitory receptors such as CD22 and PD1, which contain cytosolic immunoreceptor tyrosine based inhibiting motif item, and sometimes immunoreceptor tyrosine based switch motif items. The T cell receptor complex is made up of T cell receptor alpha and beta chain that contribute to the antigen recognition and item containing silent chain CD3 alpha, delta, and epsilon and zeta homodimer. The CD3 chain each contain items, whereas each zeta chain contain 3 items. T cell receptor ligation results in tyrosine phosphorylation of CD3 and zeta items by SRC family kinase and recruitment of ZAP70 to phosphor items, which each SH2 domains of ZAP70 binding to one phosphorylated tyrosine of items. Activated ZAP70 phosphorylates tyrosine residue on adapter, and downstream enzymes are recruited to signalosomes. Enzymes that mediate the exchange of GTP for GDP on the small g proteins such as RAS and RAC help initiate mitogen activated protein MAP kinase pathway. This pathway leads to the induction or activation of transcription factor such as JON and FOS, a component of UP transcription factor. Activation of PLC gamma 1 lead to the release of IP3 from PIP2 and from PIP2 and IP3 induces the release of calcium from intracellular store. Depletion of calcium from intracellular store facilitates the opening of calcium release activated calcium CRAC channel, or a store operating channel on the cell surface that maintain the raised intracellular calcium level. Calcium binds to calmodin and activate downstream protein including calcirone, a phosphatase that facilitates the entry of the nuclear factor of activated T cell as transcription factor into nuclear. DAG is generated in the membrane when PLC gamma release IP3 from PIP2. DAG can activate PIC6O, which among other things can contribute to NF kappa beta activation. A lipid kinase called PI3 kinase convert PIP2 to PIP3. PIP3 can recruit and activate plaque string homology PH domain containing protein to plasma membrane. PIP3 activate ITK into T cell and PTK into B cell. It can activate PDK1, a kinase that can phosphorylate a downstream kinase called ATK that mediates cell survival. Costimulatory receptor initiates signaling separately from antigen receptor, and signaling output from antigen receptor and costimulatory receptor synergize in the nucleus. The major costimulatory receptor in T cell is CD28. The B cell receptor is made up of membrane-bound Ig and associated dissolved linked Ig alpha and beta heterodimer. Both Ig alpha and beta contain item in the cytoplasmic tail. Signaling pathway linked to B cell receptor are broadly similar to signaling pathway downstream the toll-like T cell receptor. Attenuation of immune receptor signaling in B cell, T cell, and natural killer cell, among others, is mediated by inhibitory receptors that frequently contain inhibitory tyrosine containing motif item in their cytoplasmic tail and recruit phosphatase. Another important mechanism of signal attenuation involves ubiquination of signaling protein by E3 ubiquin ligase, which tag this protein for intracellular degradation. Cytokine receptor can be divided into categories based on structural features and mechanism of signaling. Many cytokine receptors use non-receptor tyrosine kinase called JAK to phosphorate the transcription factor called STAT. Which dimerize after phosphorylation, translocate to the nucleus and induce transcription target gene. Some cytokine receptors, such as those of interleukin 1, interleukin 17, and tumor necrosis factor receptor family, activate either conical or non-conical NF-kappa beta signaling. Conical NF-kappa signaling is activated downstream of many receptors, inducing tumor necrosis factor receptor family, cytokine receptor, toll-like receptor, and interleukin-1 RR family members, and antigen receptor. The pathway involved activation of IKK beta in the IKK complex, phosphorylation of IK beta inhibited by activated IKK beta, ubiquination and protosomal degradation of I kappa B alpha, and transport the NF kappa beta to the nucleus. Transforming growth factor beta bind to transforming growth factor beta RII2 II, which phosphorylated and activate the tumor growth factor, the transforming growth factor beta R1, which in turn phosphorylate and activate RSMAD. The phosphorylatory RSMAD then dimerize with co-SMAD, and this dimer enter the nucleus and stimulate transcription of target gene. Thank you very much. It has been a long reading, and hopefully you enjoyed, and all the best as you're looking ahead for chapter 8, where we will discuss about lymphocyte development and antigen receptor gene rearrangement. I think it will be a nice topic as well.